Is I audible?
Yes, sir.
Okay, fine. So this is all about the discussion of ARS prelims net exam. Okay. So before starting this, I would like to tell you something regarding this prelims that happens 2 days back, right? So please remember these things. The answers whatever I have here provided are there might be a mistakes because so what we studied and what exactly happens there is completely different, right? So there might be a slight variations in the options and sometimes answers may be also wrong. Take into consideration before making it as a final version. Okay, so whoever want to use it for the further, I suggest you to cross verify the information just before using it for the final. Okay, and the questions listed here are purely based on memory, you know it, right? So when I was writing, I almost managed to collect around 110. So along with few of the options, still I remembered how exactly like whatever exactly that we saw in the exam, I tried to display in the same way possible, but still few of the options are modified for our convenience because in the exam long descriptive kind of answers was given, right? Especially questions. So the very important one in the original exam, there is a very long descriptive kind of format of questioning was given, but here it is compromised in order to make it as a simplified version for the reference. Okay, so I totally understand all of your concern and all those things. If you ask me, the question paper is not too difficult, not too easy. I would say it is medium, because the only problem is they have given the long descriptive kind of questions, right? So as I told you earlier, if you gone through all our videos, I can guarantee you 50% of the questions you can easily answer it. Now I will now we'll see what all the things like they asked in the exams and all those things, whatever I have told you in the group, like if there is any things that we can request from our side to ASRB, like is there is any kind of request, not as a complaint but as a request. Okay, so if there is any mismatches or maybe the wrong question types or the wrong options, if there is such kind of things, please let me know at the end of the session, so that we can send a mail of request to the ASRB. Okay, now another thing is now every one of you interact with every single question. So because I have formulated around 120 along with another 20 questions without having the proper not remembered such that much proper, so that I have given in normal way. We'll just interact, we'll discuss every single question. In the meantime, if there is any mismatches also there, I will note it down and we can raise according to the requirement. If there is any problem or such things, we can raise a question. Okay, so just give me a minute. Okay, so we'll start with one by one now. So however, same way I have tried to formulate the question. So I have just given only question types. Answers I have not given here. There will be another video published, not now, maybe some 1 or 2 days later, because still there are couple of changes need to be done in terms of other questions and along with, because I have three different formats with me. The final format I need to refer, that is my own format. If I refer all those things, then I will get the proper exact thing. But now for your convenience I have made it, because if there is any mismatches, that we can raise a request to ASRB regarding the other kinds of things if there is any. Okay, so now every one of you interact with every single question, or you can also share your if there is another ideas or maybe other kinds of things, or like if you are feeling something else also, just share with me, we'll discuss and make the things happen. Okay, so the first one is the book Comparative Mycology and Biology of the Fungi, Bacteria and Mycetiosa was written by. Actually, here the question itself, they have given a comparative mycology, but it is not a comparative mycology. It is originally comparative morphology and biology of the fungi, Mycetiosa and bacteria. But in the question, they have given comparative mycology, some kind of something like that. What is the answer? Here, the first one, comparative mycology and biology of the fungi, Mycetiosa and bacteria. This book was written by whom?
Anton de Berry.
Exactly, it was written by Anton de Berry. It was written by Anton de Berry. This we have discussed in the history. Yes or not? So
Yes, sir.
yeah,
Yes,
Now we will see what are all the things that we have discussed, how many questions that we have discussed that came in exam. Anyway, so here it is supposed to be comparative morphology and biology of the fungi, but they have given comparative mycology. There is a small mistake, but still they have given options like and other things, but originally it is Anton de Barry book. Okay. Next one is virus is very smaller, generally measured in nanometer. 1 nanometer is equals to. So which means they have given virus is very smaller, that can be generally measured in nanometer, but they are also giving another point. 1 nanometer equivalents to what? 1 by 100 centimeter, 1 by 100. What is the answer for this? Third one, yes, 1 by 1000 micrometer. I think they have given mu or something like that. They have given like this only, but I remember they have given like that only. Yeah, yeah. I tried a lot of, I mean, like I spent a lot of time remembering the things and making the things like I tried to put exactly how it is in the exam, but still some mistakes might happen. You'll just let me know if there is such things will be there. I will prepare completely and I will also send you a copy to every one of you so that you can use it for the further also. Okay. And the next one, the answer is C, 1 by 1000 micrometer. Third one, virus infectivity can persist even at extreme dilutions. What is the maximum dilution that virus can still cause the disease? Now, so they have given a very long descriptive things, which means indirectly they are asking what is the maximum dilution point of a virus. What the answer is?
10 is to power.
Yes, see, 1 is to 1, probably 10 lakhs, right? Probably. So 1 is to 10 lakhs it is given. It is generally 1 is to 10 to the power of 6. Whether we have discussed this question or not in the virology.
Yes, sir.
This question also we have discussed. But the only problem is we never expected the pattern of questions or sometimes the options in this way, but somehow they have given. There is no other things like we cannot do now, but still we will just see. So this is in another what can I say that uh, uh, another model that the questions can be expected in any direction. Especially I actually thought that since it is combining with the different exams, definitely there might be a twist. So anyway, no problem, still just we'll see what all the things will be there. Anyway, answer is 1 is to this C, 1 is to 10 lakhs. The next one is the father of green revolution, Norman E. Borlaug, is actually revolutionizing and increasing the food production belonging to which field? Agronomy. See, don't see this underlines and all those things. You just tell me what you know. Agronomy, plant pathology, crop physiology, and seed technology.
Actually, question was not that one.
Please let me know what
Actually,
exactly the
question
question.
is not this one. The question is that when he got the retirement from Senate, so what position he was? As a agronomist, as a plant pathologist, as a crop physiologist, or as a seed technologist? Likewise, that question was there.
Is it the same?
Mentioning of the symmetry, yes, sir.
Okay, okay, fine. So anyway, so what might be the answer is?
Plant pathologist.
So here there is a confusion. Originally he is an agronomist. Since whatever you have raised, when he was retiring from the CIMMET, his position holding is agronomy because he is an agronomist. He got his PhD from pathology. He got another PhD from genetics and plant breeding. Which one we need to go with? Anyway, I will reform this question. Like when he was retired from the CIMMET, what is the position that he is holding, right?
Yes, sir. Yes, sir. Sir, and in question number 3 also they have mentioned TMV, sir, specially.
Special in the question number 3 also they have mentioned special TMV. Yes, sir, special.
TMV.
Okay, anyway.
TMV. So diluted point of TMV is 10 to the power of minus 6 as we have learned in
Studied,
our
yes,
classes.
exactly. Yeah. So I will also change this format. Okay. The fourth one is which one you have gone, but I have gone with plant pathology because I know he's an agronomist. I know he's a plant pathologist. I know he's a. I actually gone with. Yeah, yeah.
Because I have gone through 3 or 4 multiple classes, so plant pathology is not.
Yeah. I will show you one more thing now. Of course, I agree with you because there is a problem. I will show you. I'm just typing nothing else. Norman E. Borlaug. You just see Wikipedia, nothing else. We don't have that much time. You just see Norman E. Borlaug is an American agronomist. First point. We'll go to the next one. Education and life. He's graduated from somewhere else. Now we'll see another thing like somewhere I read pathology. I'll show you that one.
Sir, actually, he did from PhD in genetics and breeding as well as plant pathology.
That's what I'm see here.
I read from some.
Yeah, in Wikipedia itself it is there. Borlaug received BSc in Forest 1937 and PhD from plant pathology and genetics and plant breeding in the University of Minnesota. So here, which one you need to go with? We need see that since I have gone through pathology, that doesn't mean the option of pathology is right. Of course, there might be other chances also there, but originally he's an agronomist. Since the option was there, like when he was retiring from the CIMET, what position he was holding? He was holding originally agronomy, but we pathologist is considered as a plant pathologist because he's also received Nobel Prize in 1970. Also read his life stories also, but majority of the contributions he actually related it to plant pathology and as well as genetics and plant breeding, right? You might have studied this how the green revolution started, Norin 10 variety cross breeding and all those things. Purely was working on plant pathology and genetics related aspect, but still this question was there. But I cannot say which answer exactly, how what they represented and how they have mentioned here, but I have gone with this plant pathology. But the original thing, no idea. So which option they have given, what they will consider, no idea. But I have gone with the plant pathology. Originally he's an agronomist. The later things we will see. Next one, third one, fifth one. Fusarium ear rot in maize is mainly initiated through the infection and transmitted through. I think there is a small change in this question. Probably they have asked like where exactly this infection starts, right? Yes, where exactly this infection starts or something like that will be there. So originally the answer goes to generally the kind what they have asked is generally silk it is. So there might be slight variations also there, but I exactly not remember because that the questions are very lengthy way.
Sir,
Yeah,
actually, sir, option was not root. Option was stem instead of root.
instead of root, stem was there. Okay, fine, I will change it. Okay, but the originally answer is silk. Okay, so maybe the based on the way they have originally mentioned in the exam, it also slightly changes also, but I have gone through various resources. It is originally through silk. Okay, see now there is no point of looking back of your whatever you have selected there in the exam. What we are discussing, there might be a I mean, the wrong answers also will be there. Don't consider for your final evaluation because the somehow the result will come. There is nothing to look back now. We'll just discuss the things. Okay, so anyway, silk. So next one, Goa fruit canker is caused by, this is a direct question. Goa fruit canker is caused by Pathology. Now my question is whether we have discussed this one or not.
Yes, sir. Yes, sir.
Because I have sent you one diseases of crops, like list of important diseases of each and every crop was there. Na? I have sent one PDF, if I am right. In that PDF, go to Goa. There it was there. Goa fruit canker is caused by Pathology, which means this we have discussed. But since there is a lot amount of questions are there, there might be less chances of you to refer all these things. But still, this is a direct question, no indirect things. So the next one, typical size range of a bacterial cell. What is the answer? 1 to 5 micrometer long, 0.5 to.
Sir, C option.
This is the problem. I will tell you, there are books available. You can see there is a book called Alice and J. Lakshmi. In that book, it is mentioned as bacterial size ranges from 0.5 to 1 into 2 to 5 micrometer in diameter, which means it starts with 1 to 5. And since you have mentioned, there is another book that is mentioned that the bacterial size ranges from 1.5 to 2 into 3 to 5 micrometer in diameter. So which means both things will be there. Which one we need to go with? That is another confusion. Okay, since you have mentioned C, 3 to 5 micrometer long and 0.5 micrometer width, I am leaving it to you, up to you. Okay, but what I studied in one of, yeah, please.
Sir, even in KK sir, see, I have read now somewhere. Sir, even in KK Mandal, they mentioned 3 to 5 meter long, micrometer long, and 0.5 meter width.
Yeah,
KK Mandal, they mentioned this.
I will give you one more reference. If I am right, that a book name is however, we have Plant Pathology, Bible of Plant Pathology and Agrios book. For bacteriology, there is one book is called Master Gatto, Plant Bacteriology Master Gatto, right? You just go to that book, again there is a confusion. So anyway, I am leaving it to you. If it is a 3 to 5 micrometer long and 0.5 micrometer width, please refer once again and make sure what answer it is. That's why few of the things, whatever it is a confusion in many areas, it is.
KK Mandal book, we can't refer for that.
No. I can't say that. That is one of the fantastic book. There is no doubt about it. You can refer, but still, I'm telling you, please reconfirm once again. Okay. But anyway, I read even in many of the articles it is mentioned as 3 to 5 micrometer only. Okay. I'm telling you, please reconfirm once again. Okay. I'm not telling.
At few places the range was given 2 to 5 micrometers. So according to that, 2 to 5 is not anywhere. So 3 to 5 micrometer was looking suitable. And also, then I marked this.
That's okay. That's okay. That's what I'm telling.
The option was like this only, so very narrow range.
Yeah.
So that's why it got.
Yeah, I'm telling you, please refer once again. That's what I'm telling. See, if I know the answer, I can also tell you the reference with 100% accuracy. But still, there is a confusion. That's what I'm leaving it to you. Typical range of a bacterial cell. Okay. But in a couple of the books, it is also mentioned that 1 to 5 in the KK Mondal book, and there is another book, Master Gatto, and I'll also tell you one more very famous book that is also bacteriology, published in APS. Uh, uh, that book I actually forgot. That book also it is mentioned as 1 to 5 micrometer. So slight variations are there. Please do refer this one. Okay. The next one, which of the following is an example of ectomycorrhiza? I'm not sure about this question, but this question was there. Like the options were slightly, maybe the way of pattern of asking a question is slightly. I have not remembered. That's why I have striked it out. So I actually told you, please send me 10 questions, every one of you, but none of you sent. Only one or two members sent. But anyway, no problem. If still there are mistakes or persist here, no problem, you'll send me back once again. I will reform once again back, and I will send the full copy to you so that everyone can utilize it. Okay. So next one, which is not considered as angiosperm? 10, which is not considered as angiosperm? Rutaceae, Loranthaceae, Warbancaceae, Convolvulaceae. What is the answer? See, originally Rutaceae is also an angiosperm. Rutaceae is also an angiosperm, but
Sir, the may be like which of is not considered as angiospermic plant parasite, like that.
Is that line was there? Not angiospermic plant parasite. Is that line was there?
Yes, sir. Yes, sir.
Then it is Rutaceae. Then it is Rutaceae. I will tell you why. Of course, it is Rutaceae is also belonging to an angiosperm, but it is not a plant parasite. It is actually belonging to citrus-related families. So then the answer is Rutaceae. Of course, I also gone with Rutaceae only because Loranthaceae, Warbancaceae, Convolvulaceae, that we know very well. But I was a little bit confused because angiosperm means Rutaceae is also an angiosperm. But if that point, whatever the point that you have mentioned, like which of the following is not an angiospermic plant parasite, if that word will be there, then definitely the answer is Rutaceae only. Okay. There is no confusion, but generally I was a little bit confused, like why this have mentioned this Rutaceae, because Rutaceae is also an angiosperm, but since that point was there like plant parasite, so definitely it is Rutaceae only, no confusion. Okay. The next one, cryptogram of CMV. By seeing this question only, I frustrated like anything else. See, these cryptograms we are not using anymore. It was, we have omitted this in 1970s itself. See, how like the examiner, how he want to twist the questions, not to twist the questions, he was actually made the questions by in a different set of mind, in like doing something else. But see, this cryptograms,
Whatever option you gave, it's not a single option.
not a single option was there. That's right. That's right. Not a single option was there.
Because
But
acid in the last somewhere, acid also mentioned.
yes,
He was also
yes,
mentioned.
yes.
That's
That's
like that also
that's
mentioned.
that's what I'm telling. In the beginning only, I have given you disclaimer because some of the options I have given it for convenience, because such kind of things none of us remember the option because R3 D3. But if you give me some time, I will go through some of the books. There they have given full length things, but none of the options it was given in the exam. That's right. But this question was there, cryptogram of CMV, right? So this was there, but option, option I don't know, answer I don't know, but I have generated by seeing this. Yeah, please.
Sir, did they ask cucumber mosaic virus or cowpea mosaic virus?
They have given
CMV only.
they have given CMV, just CMV. Okay.
Just CMV. Okay.
Yeah, they have given. If I remember, they have given just CMV.
Options were differing only in the transmitting agents, sir. Like aphid, AP, Coleoptera, beetle, CL. AP was there, CL was there.
Fungus was there. Seed was also there. Then some mechanical was also there, like that they mentioned.
Yeah, they have mentioned. If you remember, please send me because I will just erase it and I will make another one. Okay. So this one I am leaving it to up to you because since we have not remembered the options properly, but I have made it for myself. The answer is A, R3, 6, 32, 34 like that. The answer is A, but the options is completely different from the exam. So I am leaving it to you. Okay, next one. Papaya bunchlet virus is transmitted by Leafhopper Emposca papaya, Leafhopper Emposca stevensis, again Leafhopper Emposca papaya and stevensis both. Last one Leafhopper Oraceus albicindus. What is the answer?
Both, sir, both.
Both.
Sir, both.
Yes, that's right. Whether we have discussed this question or not?
Yes, sir, discussed.
Because it was
Yes,
it was there in the virology, the two especially vectors. Originally it is not a virus, it is actually lactoferous FEB. It actually classified under FEB, but still we have discussed this one in the that section. There, if you remember properly, I will also show you. See, today it will take at least 1.5 hours if you can spend you know uh,
No problem, sir.
yeah, we will discuss a little bit
okay, sir.
detail.
Take your
Yeah.
time, sir.
So I will show you one thing because it is already there. We have discussed this one in especially the virology section. Yeah, see this virology section. Mm, yes, take a look at this one 27. So 27, this is already we discussed. I changed nothing because if you want, you'll just go back and see the videos also. Papaya bunchlet virus. It is actually not a virus. It is actually lactoferous FAB. See, Emposca papaya and Emposca stevensis, which means it is already there. Sometimes it will happen, but it is a direct question only. Okay, the answer is C. Both leafhopper, Emposca papaya and Emposca stevensis, both. Okay. The next one, sandal spike.
Sir, then what to think? If it is CMV, sir, is it cowpea or is it cucumber? If they have mentioned in abbreviated form, is it cowpea or cucumber, sir?
I think it's a cucumber mosaic virus. I feel it's cucumber mosaic virus.
CMV only, no, sir.
That's what I'm telling. Maybe it's cucumber.
For the CM, sir, for CMV they will mention COMP. Small O they will mention.
Acha, for cowpea it will be mentioned like CO.
Yes, I feel that.
Small O and B.
Yes.
Okay.
Yeah, I feel it is a cucumber mosaic virus. So they have mentioned, if I remember right, they have mentioned CMV directly. I feel it's cucumber mosaic virus.
Yes, sir. CMV only they mention.
Yeah.
So the answer will be like aphid, sir. It was clearly mentioned transmitting.
Yes.
Because difference was in transmitting vector only.
Anyway, thanks for the point because this is another point you guys need to remember. If you see every single question, there might be a hidden answer somewhere else. For example, I will also show you one more area. For example, somewhere it is mentioned as V-shaped chlorotic spots, especially it represents to black rot of cabbage. Somewhere it is mentioned in the xanthomonas, yellow color pigment. It represents xanthomonas. Like that, even though they have given a lengthy description, somewhere they have given the answers also. But we have not realized by seeing the long description. Yes. Exactly. Somewhere the only problem is.
Sir, actually for doing the hidden trick, we have to spend our time to read the whole question because hidden trick was
Exactly.
So
See,
that's why it got wasted the time.
See, there is.
Sir, we have to scroll the screen so many times down and up and down sidewise. Screen was not
because
enough.
The question was in bilingual, so it took long.
Sir, the main issue was also in attempting the question that mouse was after clicking 2-3 times some options were getting clicked. So I changed.
Yeah, this is not only problem with you. Every one of us, like by reading the question, majority of us spend a lot of time. But again, yeah, again, studying the full length question papers, then only we understand where exactly the trick is. Again, it will take definitely consumes a lot of time. With the experience, you can do it. See, this is the first time I took a lot amount of time. I took 75 minutes to finish for the first screening. Second screening, I started writing the things. Like again, I need to come back and tell you, that's why I started writing. It is actually the longest time I took. See, the only thing is with the experience you will get it because where the hidden things will be there, where exactly they have given with the experience, you will slowly get it. But anyway, there is no other choice. You need to read the questions very carefully. They need to find the things and all those things will happen. But we cannot do anything. See, it's already happened.
Because somewhere they mentioned which is the correct statement, which is the wrong statement. That's why it took a lot of time.
Exactly. That is the problem.
If they are asking wrong or right,
yes. Exactly, that is the biggest problem. That is, see, because we need to verify 2-3 times by seeing the options. Also, we need to verify whether they have asked right up right sentence or like the wrong thing that they need to mention. So that is also biggest problem, but still it is okay. See, this is a completely unique or the different question paper that whatever we have seen over the last 10 years, but it's okay. I never blame anyone, but I would say uh, uh, the question paper is still medium only. I never say it is difficult. It is still medium only. The only problem is long descriptive type. But still, there is nothing to worry about it. It is already done. Past is past. Let's hope for the best. Now we'll discuss whatever that what we can do. Okay. Next one. This is a direct question. Sandal spike is there, which is caused by fungi, bacteria, virus, cytoplasma. What it is?
Phytoplasma.
Whether we have discussed this one or not.
Yes, sir.
Yes, sir.
I have in the general plant pathology, if I remember right, I have given you like the each and every group of plant pathogen, fungi, bacteria, virus, along with the what is the first disease reported, what is the virus, and all those things. In the section of phytoplasma, it is mentioned sandal spike first reported in India by Verma 1969. If I remember right, that was there in general plant pathology. Go and take a look at sand spike is a phytoplasmal disease. In the meantime, we also discussed the same thing when we were discussing about phytoplasma in the list of important diseases and their vectors. There I have also given this sandal spike, which is also a phytoplasmal disease. But anyway, it is a direct question. Okay, phytoplasma. Next one. This is also we have discussed, but they have given in a different way. Citrus greening is one of the most dangerous disease. Originally, it is a xylem limited FEB, phloem limited FEB, bacteria, virus. How many times I have asked the examples of this FEBs, right? So which one it is? Citrus greening is a. Yes, it is. It is phloem limited, right? It is a phloem limited gram negative FEB, right? So xylem limited positive, gram negative, phloem limited gram positive, gram negative, all those things that we have discussed earlier. I hope by seeing this question you can definitely answer it. This is also we have discussed, right? Next one, bacterial chromosome. Even a UG student can answer this one. Single linear double stranded, single elongated single stranded, like single circular double stranded, single elongated. What is the answer? It is a single circular double stranded DNA. There is nothing to confuse because plasmid is also circular double stranded DNA only, but in general there is only one bacterial chromosome will be there, and it is a circular double stranded DNA. Sometimes in very you may study it somewhere else, it also look like linear, but mostly it is a circular double stranded DNA only. And in between, if there is any slight variations in the questions that is formed here also, you just tell me. I will note it down and I will make it right, and I will be sending a copy. Okay. The next one, answer is single circle double stranded DNA. Okay. Next one, which of the following is not true about prokaryotes? True nucleide absent, true nucleolus. Again, it is small confusion, but this option was there. 70th type of ribosomes are absent.
It is present. It was absent written. So option D is right.
Sir, actually, option number C, the option C was wrong because they mentioned this one membrane bound bodies were present like that was option was also there instead of true cell membrane absent. It was this option was not there.
Wait. Third one.
C is also wrong.
Wait. Just give me a minute. First come with C. What is the option C? Membrane bound?
Membrane bound
organelles present like that they mentioned.
Membrane bound organelles they present. Then now come you.
I just think there was an option.
Membrane bound organelles absent.
Wait. We will go one by one. Kailas, it is membrane bound organelles absent. Next one, please.
Membrane bound organelles absent.
Absent.
Like
Okay. Wait. The next one, please. Next one. Someone is telling.
The D option, 70th ribosome absent, but in prokaryote it is present.
That's see, they have given is not true, which means 70th ribosomes absent is not true because present. That is the answer. What I am telling is about the options. Whether the other options are exactly there. See, I am trying to copy exactly however it is there in the. See, if there is any like.
So option C was not what you mentioned. Option C is not here.
There. Okay. Yeah, I changed it. I will change that one. The answer is 70th ribosomes are absent because prokaryote 70th ribosomes are present. Since the question is not true about the prokaryotes. 70s type of ribosomes are absent is not true, because 70s type of ribosomes are present in bacteria, right? If there is any variations in the other options, please let me know. Don't be silent, because if you will be silent, I thought whatever I remember is right only. Okay, if there is any other changes in the other options?
No, sir. Membrane bound, which one? Third one mentioned was right, sir.
Okay, membrane bound organelles absent, right? Anyway, I will change that one. If there is any other changes like A and B, is fine, right? So the answer is 70F ribosomes are absent. Yeah, 70S ribosomes are absent. That is a wrong one. So the answer is D. Okay. Third one, Clistothesia. This is another direct question. See, 15th also we have discussed, but we have not discussed in this way. We have discussed in a different way, right? So anyway, coming back to the 16th one, this we have discussed multiple times. Clistothesia with multiple asci and appendages are at coiled tips. What is the answer? Uncinella. This is a direct question, because uncinella with coiled tip, multiple number of asci or clistothecial appendages with multiple number of asci are present inside, and the tips are coiled, or the tips are curled, or something like that will be there. It is actually uncinella. This is also direct question. We have discussed this one, right? The next one. So which of the following fungi comes under Paranosporiaceae? This is also direct question. There is no special discussion is required, because this we will discuss in UG itself. Pythium, Phytophthora, Albugond, Erisiphe. What is the answer? Phytophthora. See, there is no confusion. Phytophthora is always comes under Paranosporiaceae. So now in the recent classification, this Pythium is also placed under this Paranosporiaceae, but right now, according to the options, which of the following fungi comes under Paranosporiaceae? It is originally Phytophthora. Phytophthora comes under Paranosporiaceae. In the cricketal classification 2008, if you see that classification, this Paranospora is actually classified under, Phytophthora is actually classified under Paranosporiaceae. Okay, if you want, you will just go and type in the internet. This Phytophthora actually comes under Paranosporiaceae. Even in the recent thing, also it actually classified under Paranosporiaceae. No doubt. No Pythium, no Albugo, no other thing. I believe there is no option of Erisiphe, right? Is there is any option that you have remembered from this? I think there is no option of Erisiphe.
No, all the options is correct.
All the options were there. Okay, fine. Anyway, it is answer is Phytophthora. I'm telling you once again, there is nothing to feel by seeing the kind of answers or other kinds of things. You just see. Okay. Another thing, computer simulation model used for the disease forecasting of rice is Blightcast. They have given one sentence, like one statement. The next one, which expert?
Not for forecasting of rice. They mentioned late blight of potato.
They mentioned late blight of potato.
Yes, sir. And what is the expert for wheat?
Wait. They mentioned late blight. Yes, this late blight of potato, right?
Directly ask what is wheat, na sir? Why to mention that and asking this?
To confuse you. See, that is time waste for you. For them, that is a trick. You need to understand the point.
2 hours and giving this much lengthy paper.
See, that's what I am telling. We cannot raise a question, or we cannot ask them why you are asking like this, right? So we can see.
Sir, whatever the option you mentioned, that is not there in. Just plasma was not there. Only foam was there, and PSACO, that was also not there, because the somewhat options was
different. Yeah, unknowing. That's right. That's what I'm telling you. What are all the things that are some of the options that I have taken for my convenience, because we have not remembered that properly. That's why I've given here. So I believe the answer might be counselor or might not be there also. I'm telling you, this is just an prediction.
One option was casino, something was there.
Which one?
It was started from. One option was casino, something was there.
Casino.
Some like that question option was there.
We exactly need what exactly the option it is there. Probably it is maybe counselor, may not be. I'm telling you once again, this is, don't take it into the final version. Maybe or may not be. If you remember the option, please send me. I will reshaping it and I will sending you back. This is a little bit doubted question. Okay. So directly asking their expert system. What expert system we have studied is related to this wheat disease. There is an expert system called EpiPre. There is another plant DS, soybean related. They may ask that kind of things, but still they have a, for confusing us, they have asked all these things. But still it is okay. Just if you find somewhere else, please let me know, because I have just searched. That's what I am telling.
I think this question was in your book, sir.
Neha. Yeah. I do mentioned, but I do mentioned EpiPre. I do mention. I remember.
No, counselor, I read, sir. In your book, from your book, objective only.
Okay, I'll just give me 1 minute. I will go back and I will see, because I know I wrote this one because in the epidemiology I think it will be there. Probably somewhere it will be there. I'll show you one more thing. Yes, you'll just see here. Just give me a minute. Yeah, see
Sir,
here.
in the book MK Adam, page number 195, question number 26.
It is.
There was given the weight is the the counselor is the weight expert system.
There is no doubt. Then go with counselor. Okay. So yeah, in my book also probably I have mentioned somewhere else, but what I have given here, it is like Epipre is one of the system preventing the spring of winter. It's something like that I have given here, but I believe it might be counselor. That's why I have given. I'll just underlined it, but it is not sure. If it was there in the MK Yadav sir book, so then I suggest you please go with this. Okay. If you remember the other options also, please let me know. Okay, I will update this one in the upcoming editions. Okay. The next one, biocontrol agent for root rot causing diseases. Root rot disease caused by Rhizoctonia solani. So what answer it is? Pseudomonas fluorescens, Trichoderma viride, Glycladium varians, and Chaetomium globosum. I think the last two options, I don't know whether it was there or not, like Glycladium varians or Chaetomium globosum, but these two options were there. Pseudomonas fluorescens.
No, sir. Bacillus subtilis was there.
Just
Bacillus subtilis
wait,
was
wait, wait,
there,
wait, 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 wait. Bacillus was there. Okay, okay. Bacillus subtilis was there. Okay. Did you remember the another option D? So okay, anyway, what might be the okay? Anyway, leave it.
Chaetoplima present, sir.
Chitom is also there. Okay, fine. Then D is right. The C is supposed to be changed. Okay, then I will do it. What is the answer?
Trichoderma.
Exactly, trichoderma. There is no confusion because Rhizoctonia is a soil-borne pathogen. All the soil-borne pathogens are effectively managed by the application of trichoderma along with farmyard manure. We have studied somewhere else in the theoretical material, right? So for this trichoderma, for all the soil-borne, you know it, Rhizoctonia, Macrophomina, Sclerotium, Sclerotinia, all are soil-borne pathogens. They are very effectively managed by the application of trichoderma. So the answer is trichoderma only. Okay, there is no doubt. Okay. So the next one, potato scab, Streptomyces scabis, transmission from one area to new area or something like that. They have given with how it is actually spreading or something like that. They have given like.
Sir, in one option, they mentioned bacteria is present in vascular bundles of the potato like that.
Yes, sir. Yes, sir.
Wait. See.
Vascular bundle was there.
Wait.
From tubers.
Wait. First of all, we will evaluate the question because I have a doubt on the question whether they have asked the same thing.
This streptomyces not be mentioned. They mentioned Bangle blight of potato is transmitted from one area to new area.
It is bangle blight or potato scab. Are you sure?
Yes, sir. No, sir. It's Bangle blight. Still I remember. No, sir. Bangle blight. Bangle blight is transmitted from one area to another area.
Wait. 100% sure. Okay, whether it is a bangle blight or potato scab, please let me know. Again, you just recall the things, but I feel that might be somewhere related to this potato scab. If you are 100% sure.
No, sir. Scab portion was another, but the transmission.
Yes, sir. It was Bengal blight. I think.
Okay.
Questions are present.
See, that was there. That was there. Somewhere I have given this question also.
Both question was there, but Bangle blight is for this question transmission of that for Bangle blight they mentioned, and that vascular bundle something was question option was also there.
Correct. Yes.
Oh, just a minute. Through vascular bundles. What is that option? Through vascular bundles or what?
No, sir. Bacteria is present in the vascular bundles of the potato.
Wait.
Question is transmission of Bangle blight from one area to another area.
Present in vascular bundles. Wait. Bundles. I'll change this option. What about the remaining options? That is okay.
Sir, I think second option was also not there.
Second option is.
Second option was not there because they mentioned soil. The bacteria is present in the soil like that they mentioned.
Bacteria.
No, B option was there. B option was there present. The B option is right.
The B option was there.
B option.
Third, seed and soil is not there. Only soil was there. Seed they mentioned. They didn't mention in seed. They mentioned only soil.
Only soil.
I think that was survival of the Bangle blight pathogen, and the option were acidic soil, tuber.
Yes, sir. Acidic soil was there. Vascular bundle.
Wait. wait, I will note it down. Come again one by one. The first one is bacteria present in the vascular bundles. That option was there. Fine. The second one is bacteria present in the soil or actually survive in the soil. That is option also there. Another option, inoculum, bacteria present in the acidic soil. Okay, fine. Acidic soil. That's why I told you to please remember 10-10. If you might have done the job become very easy, but anyway, still no problem. Tell me now, the bacteria present in the acidic soil, that is okay. And another thing, inoculum present in the surface of tubers. This option was there, I feel.
Yes, sir, this was present.
Yeah.
Yes, on the tubers.
Definitely, yeah, that was there. And the one more option, like bacteria on the lenticels, this option was there, or
No, sir, I don't think it was there.
it was not there, right? So, but anyway, no problem. What is the what might be the answer?
Sir, I have written B.
Vascular bundle.
You have written. It's okay, fine. Please, I'm leaving it to you only because.
Sir, actually it's a vascular bundle because they are asking about the Bangle blight of potato that is caused by Ralstonia.
Okay, anyway.
So they are asking about the transmission.
That's
Yes, sir, they were asking about transmission.
Yes, sir, they are asking about the transmission.
That's what I'm telling. I'm leaving it to you because if I am 100% sure, I might have told you the answer. That's why I'm leaving it to you. Okay, because again, since we have not remembered 100% properly. Hot water. Just 1 minute. Hot water. Just కొంచెం బాగా వేడి చేయి మీడియం. Yeah, please. So anyway, since there is a gap in the question, that's why I'm not giving the whatever the answers I'm not giving. I'm not 100% sure. I'm leaving it to you only. Okay. But one thing I remember that there.
Based on your experience, what might be the answer?
The might be vascular bundle. Might be. I'm not sure because the one thing is like since they have mentioned another transmission is through or like see this bangle blight of potato. Generally, what is the propagation we used to propagate through seeds like the tubers and all those things will be there because they have also mentioned one more point from one area to another area how it is actually spreading, right? Through vascular bundles, like maybe through potato seed tubers also there might be a chance. Other kinds of
Sir, option was the bacteria present inside the vascular bundles of the potato seed. That option was.
Yeah, that was there, but that's what I'm telling. I'm leaving it to you.
For now, we can skip this question.
We'll go to further thing. Yeah. See, anyway, no. See, yeah, no problem. Discussing is a good, like we'll get some new points also. But anyway, next one. Which of the following disease is favored by? See, they might have asked the direct question. How much long they have stretched? Which of the following disease is favored by night temperature 20, day temperature 30? Did we study this kind of things anywhere? Not even in our UG, PG, nowhere else, right? But still they have asked this kind of things. Night temperature 20, day temperature 30, followed by leaf wetness period, prolonged leaf wetness period of 14 hours of light and 10 hours of dark, helping for the infection of which of the following disease. The options are 100% clear. Blast of rice, bacterial leaf blight of rice, brown leaf spot of rice, bacterial leaf sheath of rice. What is the answer?
No, sir, the leaf sheath was not there. Leaf streak was there.
Leaf, sorry, it is actually leaf streak only. I thought I have typed the same, but anyway, no problem. 21 leaf streak. Okay, leaf streak. What is the answer?
Bacterial blight.
It is a blast of rice. It is a blast of rice. Okay, the answer is blast of rice. No.
For blast, maximum temperature they need is 20, but they are saying 30-degree temperature.
That is a morning temperature.
But
See. Here, see, that is a morning temperature. Night temperature 20, morning temperature 30, leaf wetness period 14 hours of light and 10 hours of dark. So originally they have mentioned in a tricky way. You just type the same thing and there is a right side, there will be a cursor called books. You will redirect to the books. There are a couple of books they have mentioned the same point. So it is actually blast of rice. Okay, if you have encountered other kinds of point, just send me back and we'll discuss. Okay, but originally the answer is blast of rice. There is no doubt. Okay. See, what we are discussing is not 100% right or true. I am saying, as I told you in the disclaimer itself, there might be a chances of things like errors also may happen. But I am telling with my knowledge, I am telling like, okay. So this is, I am telling means that doesn't means that is 100% true. There might be errors also will happen, that I am telling. Okay. The next one, which of the following fungal, next 22, which of the following fungal spore producing an alternate light between dark and periods known as circadian rhythmic phenomena 24 hours by, whether we have discussed this question or not? Circadian rhythmic phenomena or 24 hours biological fungi. What is the answer? It is Pilobolus. There is no confusion. 100% it is Pilobolus only. I think the options are slightly changed for our convenience. If you have remembered, please let me know the options. But the answer is without any doubt, it is a Pilobolus, no doubt. Okay. Next one. Yeah, this is another thing. They have given something like soil regarding or related to the soil solarization kind of thing, and along with they have given a statement kind of thing. Like statement one is soil radiation actually increases the soil temperature, where this polythene sheet traps the solar light and raises the soil temperature. The second statement is greenhouse effect of solar radiation increases the soil temperature through polythene sheet, something like that. They have given two statements, but the options I have not remembered. That's why I left it free. If you remember, send me later. Now it is not time to discuss all those things. If you remember regarding to question number 23, please take a pen, note it down all these questions. Let me know once after it is
was said that both options are true and this justifies option one.
Yeah.
This was like that.
Here it is given both options are true only, but whatever the answers that I have given, I am not sure. That's why I have left it free. But because the both statement it is given is right only. Okay. The next one, Aspergillus is a human pathogen, mainly infect which of the following organs of human body? Eye, lungs, heart, kidney. Yeah, it is primarily targeting lungs, but it also infects eyes also. But here in this case, answer is lungs also. If it was given it as bones, eyes, lungs, other kinds of things, because this Aspergillus is one of the very problematic pathogen, especially in the bone marrow transplanting surgery. So since the bones, all those things was not there, the answer goes to lungs, because whenever we are like generally, yeah, please.
There was an option as the place of kidney, that was stomach.
Yes, sir, it was stomach.
Wait. In place of kidney, it is stomach. Okay, fine. Okay, I will replace it. Okay. But answer is lungs only. Okay. Anyway, the next one, 25. The mode of action of streptomycin producing taxotomin is mainly useful for tissue disintegration or maceration. It is tissue maceration, right? Not disintegration, right?
Yes, sir.
It is tissue maceration or tissue disintegration?
Maceration, sir.
Oh, maceration was there. Okay, tissue maceration was there. Okay, fine. Second one is pathogenicity, enters into the plants, killing of the.
Not only pathogenicity, I think there mentioned
Yes, sir. Yes, sir. Pathogenicity was not there.
Wait. Come one by one. What is this?
Increase in pathogenicity or related.
B, increase in pathogenicity. Okay, increase in pathogenicity. Okay,
Sir,
fine.
decrease in pathogenicity actually.
It is decrease and increase in.
Decrease, sir.
Decrease, sir.
Decrease in pathogenicity. Now the answer is very clear. Enters into third one, enters into the plant cell, killing of the plant cell. What? See, C and option D is added for the convenience. What might be the answer? So if in this case, if you have mentioned that it decrease in pathogenicity, definitely the answer is tissue disintegration. Yes, definitely the answer is tissue maceration. I have striked it out or underlined it just because taxotomin is also one of the phytotoxin, generally actually helpful or plays very important role in pathogenicity. But since you have mentioned this point, my answer is very clear. It is a tissue maceration. So because this taxotomin is also one of the important factor in pathogenicity. So since if it is a decrease in, so it is very clear it is a tissue maceration. Okay. So C and D options were added for the convenience. If you know, please let me know the other things. Please take a pen and paper. At least please do me for now, because whatever the contribution you are doing, it is for the not only for us, for also for the future, right? So the options also, if you try to make it as exactly however it is there, it will be helpful for the upcoming one. Okay. The next one, Kingsby medium is a synthetic, semi-synthetic, mixed media, natural media. What it is?
Synthetic, sir.
It is it is a synthetic one. Okay, some of the articles it is mentioned as semi-synthetic also, but originally it is a synthetic. Okay. The next one, which of the following is right for the management of wheat loose smut? They have given like use of healthy seed material, hot water for 53 degrees Celsius for 30 minutes, use of carboxin group for seed treatment, use of triazole fungicides. So they have given is A and B only right, A, C right, A, B, C right, A and D is right. What might be last one?
Option was also there. All are correct.
All the above, right. Remember, all the above. Yes, that's right. I remembered it. Just now tell me.
That is only the answer. All the above is the answer.
No, sir. ABC only.
I feel ABC is the answer. Yeah, I feel because this triazole fungicides we never use it.
ABC only.
Yeah. See, that's what I'm. See, don't worry. What I'm telling is just my view only. I'm telling. It is not 100% sure. I feel it is ABC because this triazole fungicides we are not using for smut related diseases. Okay. So I feel it may be ABC, maybe all the above also, but I feel what I have gone with is ABC. That doesn't mean that is right. Okay, I'm telling you maybe ABC or might not be also. Okay.
Actually, trizoles mostly used for rust, not for smut, mostly for foliar.
Yeah, that's what I'm
spray and
telling.
like that, not for
Might be, that's what I'm telling. Maybe or may not be also. What I know is, according to my knowledge, there is no special case that this triazole fungicides were using for this smut and bunt disease, especially loose smut related things. We mostly study carboxyn, and there are millions of articles are there purely on the carboxyn, but I never studied anywhere related to triazole. That's what I'm telling. Might be ABC. Okay, we'll leave it as such. Okay, maybe all the above also, but I feel it may be ABC, but I have gone with ABC. That's what I'm telling. Okay. So anyway, the next one, this is a direct question. This question, whether we have discussed it, discussed in the sense this is there in one of the exam.
Yes, sir.
This is there in one of the exam. I'm very sure. In one of the,
actually one option was in area.
Yes, sir. Yes.
wait. Your voice is not clear. Please come again. Kailash,
He's saying that one option was area gram.
area gram. Okay.
Yes.
One option, area gram. Okay. So the what might be the answer?
Field runner.
Field runners.
runner.
Because this is a direct question, and we have discussed the same thing in our daily tests also, like weekly test or monthly test, I think so. The next one, yeah, this is one of the fantastic question. There in somewhere the answer was hidden. Actually, the smut of the floral pods after the emergence, few of the grains are rotted and they produce black powdery mass of spores with emitting a bad odor from the infected plants. Is a
Kernel
characteristic.
bunt.
It is a kernel bunt. See here.
Actually, hill bunt was also there. Option was hill bunt is also there, and one was also there, sir.
Okay, one option hill bunt was there. Okay.
The one is hill bunt option is there, and another one is some uh, smart or something was there.
Okay, fine. That's okay. No problem. What is the answer?
Kernel bunt.
Here it is a
Dimethylamine was
yes.
it.
Here 2 points are there. First one, they have given only very few grains are infected, because the kernel bunt is also called as partial bunt. They have mentioned one hint, that is only few grains are infected. First point. The second one, instead of mentioning trimethylamine, they have mentioned a bad word. This is also another representation of kernel bunt. So the answer definitely might be the kernel bunt. Okay. The next one, recent.
So what is the symptoms in hill bunt? Can you tell me, sir?
See, for all the bunt, the symptoms are most common. Like all the infected grains are converted into black color powdery masses only. There is no variations in the in this bunt. Okay, so mostly all the bunt, you can take all the bunts, infected grains, wherever the grains or inflorescence is infected, they convert into black color powdery mass only. Maybe twisting or other kinds of things also might be there. But as far of my knowledge, like smut, all smuts or the bunts, mostly they are converted or replaces the grains into black color charcoal powders only. But since they have mentioned only few grains are emitted other kinds of colors, so I believe it is goes to kernel bunt only. For further details you may refer the other things, but I feel it is a kernel bunt. Not feel, it is a kernel bunt only. Okay. The next one, recent classification of fungi. Read the question very carefully. Recent classification of fungi with molecular phylogeny is based on assembling fungal tree of life, based on the dictionary of fungi, based on taxonomy, based on.
No, sir. Option was multi-genome sequence is also there. Multi-genome sequence.
There is an wait. There is another option, multi-genome sequence.
Analysis.
Multi-genome sequencing analysis, right?
Multi-gene analysis.
Multi-gene analysis.
Yes,
okay.
Sir, multi-gene sequence analysis.
Okay, what is the answer?
Multi-gene sequence analysis.
No, it is after. It is definitely after.
Actually, after is a project, and in which they did because they are asking what is the basis. They didn't ask based on. They asked the basis.
See,
they are asking the basis.
They have wait. They have mentioned the molecular phylogeny. See, whatever it is, multi-gene analysis and all those things, this assembling fungal tree of life is actually based on what?
No, sir, molecular phylogeny they didn't mention. They didn't mention only recent classification of fungi on phylogeny. On what basis?
I am 100. Wait. I am 100% sure. I am 100% sure they have mentioned molecular phylogeny because I read this question 3 times. They have mentioned molecular phylogeny, 100% sure, because I feel anyway. See here, it is not for fight. I'm just telling because I have a carbon copy of this question because I have just carbon copied all these questions. This is yet to refer, but I still remembered because I read this question 3 times. Molecular phylogeny is also there. I feel it is after, but there is no point of fighting here. Maybe multi-gene sequence, multi-gene analysis also might be the answer. But as far as my knowledge, because regarding this project I have read, that's why I feel based on this molecular phylogeny, probably maybe after may not be also because I have gone with after. That's what I am telling. See, I am gone with after, that doesn't means the answer is after. I'm generally telling maybe whatever you have mentioned multi-gene analysis also there, but since you raised a point, but this molecular phylogeny is there in the option.
Because of the project, but
Kailas, wait. I can't hear you properly. Is it only for me or for everyone? I can't hear you properly. Come again. Is it audible? Is it audible or not? Just wait. Give me a minute. Just give me a minute. Yeah. Is it audible, right? Yeah. You tell me now. You're telling something?
Sir, actually, I just want to know in the AFTAL project whether they use multi-gene sequence or not. That's
So
only
they used it. That's what I'm telling. What? Come again?
Sir, multigene was not used in all cases. It was for resolving some species complex here and there.
Yeah.
But the actual classification was it depend on after only.
Exactly, that's what I am telling. See, because in the 10th classification, whatever the dictionary of fungi, it is purely based on the molecular phylogeny, that is purely based on the after what I read somewhere else. Maybe the option is also wrong or right, something else, but I feel since they have yeah, please tell.
Hello, sir, I just want to know that whether they use multi-gene sequence in AFTAL or not.
Which means they see for targeting they for doing or sequencing all those things, they do use different regions or the different other kinds of things also will be there. But so in order to make or this classification all those things, what exactly they were following is this after only. Okay, so I see anyway, the options all are there, but I feel it might be after all. Like you can also just refer the other sources and let me know if there is in other things, but I feel it is after all, since she has also mentioned not all the cases like multi-gene sequences can be used. So you just think about it, like if there is in other resources also, please let me know. For further details we'll discuss regarding this. It is already 1 hour, right? So we have only discussed just 30. Okay, so the next one, which of the following is not true about the virus infection? Causes yellowing in plant, reducing the chlorophyll content, reduces the respiration rate, reduces the photosynthesis activity. I don't know whether those all options are not, there or not. So this question was there, right? Like there is a question like which of the following is not true about the virus infection? So which is not true about the virus infection? Because it causes the yellowing in plant, that is a true. Reduces the chlorophyll content, that is also true. Yes, C option, because when the virus infects, actually respiration rate increases, not decreases. So might be the answer is C. Respiration rate is reduces, that is the wrong thing, because reduces the photosynthetic activity, chlorophyll causes yellowing. I think all these options was right, but this option was not exactly related to this virus. So it is not true, since they mentioned, probably it is answer C. Okay, so 32, mocha wilt of banana is caused. This is also direct question, we have discussed it many times, right? Mocha wilt of banana is caused by Ralstonia solanacearum. No confusion, Ralstonia solanacearum. Again, the which of the following is wrongly matched? Grain smut. Mocha wilt of banana. Just give me a minute. 32 Fusarium was there. Okay, one of the option. Okay, fine. The next one, which of the following is wrongly matched? Grain smut, head smut, loose smut, long smut. Just by seeing the option, please let me know what is the answer. It is actually grain set. We also discussed the same thing. Like we'll start with the grain, head, long, loose, long, like that. Like 10 years, 2 years, 4, 2 again. Like 10 years in 10 years, 1 to 10th class up to again 2. Like the amount of the seed, like how many years it will serve, and all those things what we have discussed, right? Along with the pathogens also. But here two things that I have given. Grain smut is not caused by Sporosorium releans. It is actually caused by Sporosorium sorgi. This is the wrong pair. Okay. The next one, irregularly dull yellow blotches that appears on the edges of the leaves. See, these are all not necessary. They have mentioned one point. V-shaped yellow yellow lesions that appears on the leaf margins. So the answer is black rot of cabbage. In the black rot of cabbage only where we can see V-shaped chlorotic spots or V-shaped chlorotic margins. So that is the important feature of this black rot of cabbage. Okay, the answer is black rot. Next one, cabbage yellows is caused by?
Fusarium, sir.
Yes, mostly yellows are caused by phytoplasmal disease, but there are few yellows like cashew yellows, uh, uh, cabbage yellows. There is another one yellows in the chili, if I am right. There are just 2-3 yellows which is caused by this Fusarium. Okay, in this case, cabbage yellows is caused by Fusarium oxysporum pharma species conglutinus, because this is a direct question, but I would say this is also a direct question. There is no new thing. Yeah, please.
Sir, I think viroid is also there in option.
Viroid was there in the option. Huh? Give me
Yes, sir.
35. One option, viroid, right? Okay, fine. Next one. Which of the following, 36, which of the following, just a minute. Yeah, which of the following infected plant parts transformed to floral parts into green leafy structures with which's broom makes the flower sterile? Yellows, mosaic, phyllodes, anation. This is also a direct question. You know it. What is the answer? Phyllodes, yes. So this is a direct question. So Sam phyllodes, especially you can see phyllodes is the answer. Okay, this is also a direct question. 37. Name the book that was written by G.J. Butler. This is also a direct question. E.J. Butler. Fungi and diseases in plants, right? Option A. Fungi and diseases in plants, that was written by E.J. Butler. This is also a direct question, right? So another thing, match the following. See, this is purely leaving to you. I actually made the same option, however, it was there. First one, grapes, rose, citrus, chili, along with the options also again. I will be giving you 10 seconds. Go through and let me know. Just 10 seconds.
Option A.
What about others?
I think instead of chili, okra was there.
Okra?
No, chili only, sir.
Chili only. I think it's chili only. See, I think it's chili only. I feel all the options are there. So the answer is A. So, yeah. So grapes. Wait. Grapes.
Chili was not there, sir.
Chili was not there.
Chili is not.
Chili was there.
I think.
I feel.
No, chili was there.
I feel chili was there. Yeah, chili was there somewhere else. But anyway, no confusion. So the answer is A. See, which means grapes. See, this kind of things. The only thing is you need to match exactly. Then again you need to revert.
Sir, there was papaya in option.
Papaya.
Yes.
Yes, sir. Yes, sir.
Papaya was there.
Yes, sir, papaya.
Instead of chili, there was papaya. Okay, anyway, fine. I thought might be chili was there, but still okay, no problem. We'll take it out the chili. So we'll see if you see this one, the answer probably this one, like A related to Ansonella necator, rose, Parotheca panosa. So this one, see, this you know it. Just I'm telling you. Again, next one, match the following. One is disorder and nutrient deficiency. Again, you got 10-15 seconds.
This option was not that much because in some way they mentioned nitrogen deficiency with water deficiency.
That is that is in
uh,
another case. That is in another case. That is
like
also there. Yeah, that is in another case. That is also there. If I am right, probably maybe new. If I am right, maybe air pollutant at somewhere and something like that. It will be there. But anyway, somehow.
Sir, air pollutant was also in this also was there.
But anyway, in this case, like if you see this buttoning is generally, if I am right,
Option was first option, right, sir?
I feel it might be the second one. Actually, buttoning is a nitrogen related thing. Hollow stem is related to boron thing. Chyra is actually
Yes,
zinc
sir.
deficiency. Viptyl is mol.
Yes, sir.
Probably the second thing. Anyway, there is no
Sir,
confusion.
there was an option Browning actually was there also.
Wait. There is another option called browning. Okay, fine. I will re-refer.
Yes, sir, there was browning.
Browning.
And that was due to boron deficiency.
Okay.
Yes, sir. Browning and Holocene both are there.
Browning and
In separate way, sir, but at the place of A like Browning and second like Holocene.
no problem. You will just tell me whatever you remembered, because I have one carbon copy that yet to be referred. Once that was done, we will get the exact
Sir,
clarity.
hollow stem in bracket they mention Browning, or otherwise the slash they mention Browning.
Hollow stem
Nothing was
in bracket they have mentioned. Anyway, no problem. You'll just tell me whatever you remember, we will see it. But anyway, it is related to this B, okay? Like matching after match. Yeah, go through this one. This is a, see, there are 5-6 questions, or I would say around 8 to 10 questions was asked in related to history. But the only problem is, see, all these 4 questions, they have given only 1 mark. In such way it was there. But anyway, no problem. We'll just go through and let me know. So what might be the answer? 10 seconds.
First one.
Which one?
No. Third one.
It is a third one. Okay, actually PA Michelli, yeah, PA Michelli Nova Plantarum genera. CH Pursoon, it is Synopsis methodica fungorum. EM Fries Stoma mycologicum. Butler, fungi and disease in plants. So originally it is option 3. Okay, next one, air pollutants, pan, ozone. So again, no wasting time, I will tell you if I am right. Pan, which means a silver leaf, so which means 4. There is only
So
one
the
4.
last option.
Yeah, last option. Yeah, in that way, see, rather than wasting time, yeah, please.
Just by looking one option, we can solve that.
Not all the time. Please remember, not all the time. Okay, some cases. Yeah, please.
Sir, in last question number 40, along with the book's name, they mention one more contribution of those scientists.
What contribution?
So one more contribution was also there, because in this one match the column, the book as well as one more contribution, means that option was big, just like synopsis methodica fungorum, and one more contribution of whatever the person.
But
like
that is okay. But Synopsis methodica fungorum is related to CH Pursoon only. If might have, like, see, this is for our discussion only. For I am telling you in exam point of how we need to done. Like for example, if you see this option Synopsis methodica fungorum, without any further hesitation, you can directly redirect to here. That's what I am telling. But anyway, if there is any further thing, I will look into it. Okay. Anyway, next one, air pollution. See, by seeing the only one thing, pan, silver leaf, there is in the other 3 options, there is no pan here. So definitely the answer is 4th one. It is not in all the cases. To save the time, you may follow this kind of thing. Okay. Next one, which of the following is rust disease? Fig, bear, grapes. No, again, this kind of options was there. What might be the answer? Like without wasting time, I will tell you. Probably the fig is generally, if I am right, Asium Physi. So option 2. So probably it is 2.
Asidium Physi.
Yeah, probably it is option 2. Yeah.
In this Pacospora GGP also there, Pacospora Gigicola was also there.
Yeah, one more. Pacospora?
Pacospora Gigicola was also there.
Wait. Pacospora Gigicola. Okay. Gigicola. Okay, fine. Anyway.
And the peach and plum rust also was there.
Peach and plum rust.
Yes, sir.
Peach and plum rust. Okay, fine. Now we got maybe peach and plum rust. Now the options may changes. I will look into it. Okay. Next one. So molds they have given: gray mold, citrus mold, pink mold, green mold, something like that they have given. So anyway, without wasting time, I will tell you if I am right. Gray mold, it will directly redirect it to botrytis, citrus mold, penicillium, pink
They
mold.
mentioned the gray mold, blue and green mold. There was pink and red mold.
Wait. There is one option called blue mold, right? Just give me a minute. Blue mold.
No, sir. Actually, they mentioned like this: gray mold, green and blue mold, citrus, sour citrus mold, sour citrus, and one is pink and red mold. Like that option was.
See, they have given only 4 things, not 5 things will be there. You're telling 5, I think. But anyway, no problem. Wait, gray. Wait. First one is gray, second one is blue, third one is sour rot citrus.
No, sir. One is gray mold, second is gray and blue mold.
Gray
Third one
and
is pink and red mold. Fourth one is
pink and red mold.
hello.
Yeah, please.
Sir, one is gray mold, second is blue and green mold, third one is citrus sour rot something was there, and fourth is pink and red mold.
Citrus sour rot. Okay, I will revise this one by saying it. Citrus sour rot, right? Gray mold, blue and green mold, pink or red mold, citrus sour rot will be there. Okay, this is also one of the question was there, right?
Option geotrichum was also there.
Geotrichum. Okay, in the options, geotrichum was there. Okay.
Yes, sir. Instead of graphium, geotrichum was there.
Okay, fine. Geotrichum was there instead of graphene. Okay, I will look into this one. Okay, the next one. This mold. Yeah, I think this is another thing. Like it is actually not a mold; it is actually a pathogen name. Like a bud rot or fruit rot of chili, root rot of citrus.
Sir, actually, this is for this they didn't ask math. They asked which one is wrong. Which one is wrong? Like that they asked for this question.
So for this question, they have gone which one is wrong, right? So I think they have given.
Tomato.
That's right. Bud rot of tomato. That's right. One thing they have given. Bud rot of tomato. Okay, fine. I think this is completely unique.
Tomato, they mentioned Phytophthora citrofthora. They mentioned for matching bud rot of this one. Bud rot of tomato. They mentioned Phytophthora citrofthora, and another one is which one is there, sir? Stem or fruit rot of this one. Coconut was also there.
Every one of you, please do one thing for me. Take a screenshot and please rechange it because for me it becoming a heavy one. But anyway, no problem. Just take a screenshot. If there is another things, please let me know also. Okay. Next one, matching the scientists with fungicide discovered. Mellard, Tisdale and Williams, Borlaug, Diamond, Marsh. I think another other kind of things is also there, right? By seeing the options only, we'll get it. The next one, this will be there. ZNF, TAL and CRISPR-Cas. But this thing, what they have given in one tabular column, I couldn't able to recall it. But this one is their finger, CRNA, DRN, transcript like activator will be there. But there is one column, I think they have given something else like required.
Protein needed to be synthesized like that.
Yeah, protein made it to be synthesized.
And all those are same, sir. Like same sentence.
In first option,
Yes, exactly. That's right.
For example, protein. So therefore, protein is to be synthesized, and for talent and CRISPR, it was like it doesn't need to be synthesized.
Yes. Yeah, they are given in that way. Please take a screenshot of this and please make me this question and send.
First option, meganucleases was given, sir.
ZNF, first option.
Yes.
ZNF, meganucleus. Yeah, that's what I told you. At least, please, if you send, that will be helpful for me. Okay. So at least take a screenshot, and at least if now also if you send, it will be really helpful, and I'll make another video. Like in a manual way, so that will also will be posted. So without a further discussion, that will be helpful. Okay. So this is one question will be there. Please, make the changes, whatever the wrong things will be there. Okay. The next one, this is another thing like pathotype, biotype, physiotype, stereotype is also kind of one kind of thing will be there. Another thing, hybrid or half-free moral, common moral, delicious morals or nettles, other thing will be. What might be the option? A, B, A, B, C, A, C, all the above?
All the above.
Might be all the above because verpa generally not edible, but the species whatever they have mentioned. Yeah. If I am right, the species verpa bomica. This might be. So there is an also chance of the last option also. All the above also there is a chance because this verpa bomica it is also one of the. Yeah, please.
One was that one also there.
1, 2, 3.
No, sir. It is right, which is mentioned here. A, B is correct, and A, B, C is correct.
Yeah,
A, B was correct.
I think they have given in this way only. this is a little bit one, because every single species here it is mentioned as edible only, including the verpa vomica, because when you cook it will become edible. If you don't know how to cook it, it will become poison.
The verba after cooking.
Yes,
It
yes,
is written in book only, na?
yeah,
After cooking it is edible.
yeah. Not only this one, there is another thing, helvella also, you can have it after cooking. So if you cook properly, it is edible. So the problem is all these species are edible only. Here it is mentioned A, B, A, B, C, A, C, all the above it is mentioned. Little bit doubted about, it is up to you only. Okay. Next one, chestnut blight of caused by Cryphonecteria parasitica is introduced to the North America from USA to Japan or China, USA to United States. Like what might be the answer? Like there is
Europe to USA was not there in option.
Europe to
Europe to USA was not there in option.
hey, every one of you, please do one thing. At least now take a screenshot and revise the options and send me back. So I will tell you whatever I know, because this chestnut blight, you know it. It is a very problematic pathogen in USA in 1904. It was first appeared in USA in 1904. It is believed to be introduced from China. It is believed to be introduced from China before 1904, or before that. It is believed to be introduced from China, but the question still, like whether they have asked introduced or something else, it is a little bit doubted about. If you remember this question number 50, please let me know and send it to me. Okay. The next one, choose the correct statement. Something they have given like magnification and resolution, or resolution depends on the wavelength, something like there is one question. If you know, again you make it. Okay. In ELISA, what is the correct order of adding? This you can answer it. This is a direct question. What is the correct order of adding of ELISA, like antibodies?
There were like option like common antigen also.
Common antigen. Okay, fine. Anyway,
Yes, sir.
that will make it. You'll do one thing. You'll just tell me the answer. What might be the antigen antibody conjugate, antigen antibody, something like that.
Option A.
Option A. Originally, it is actually, because when initially we will quote with antigen, then antibody is added. Last we can conjugate it, right? So anyway, if there is any changes, please mute your mic. Yeah, mute your mic. So when you are answering, just okay, without any further disturbance. Okay. Anyway, the answer is this was option was there. If there is any changes in the option, please send from your end. I will be very happy. Okay. The next one, which of the following cannot be used for the identification of defense gene expressions? Mutations were there, micro ARS, PCR, PTGS were there. Maybe PTGS, I'm not sure. RNA is also was there, right?
Correct.
Instead of PCR, RNA.
Instead of PCR, RNA was there. Okay, fine. Maybe PTJ. I think maybe I'm not sure. I'm telling once again, maybe or may not be also. This is one of the question. The next one, which gene doesn't follow gene for gene hypothesis? HM1, N1, XA21, and PTO. What might be the answer?
I think sir XA20 because it is recessive one.
No. It is actually HM1, because the HM1, the gene. Yeah. Yeah, all the N1, XA21, P2O, PTO, all this follows the gene for a gene hypothesis, except this HO1. It is actually the toxin, actually detoxifying kind of things which is produced in this interaction. So I don't think it will follow gene for gene interaction. But I have cross-checked this one. This XA21 follows, because I also wrongly mentioned XA21 only. But later coming back and I referred the article, then only I realized it is actually HM1. Okay. So probably it is HM1. XA21 follows, because if you want, you will just go to the book, like in my book also will just say XA21 is also there in the gene for gene hypothesis, or the interaction or the kind of things, AVR things that was produced, where this XA21 is also there. Okay, it is actually HA21. Sorry, HM1. Okay. So the next one, most important receptors in the plant pathogen. I have not remembered this one properly also. Please, if you know.
The option you left was protein kinases.
Protein kinases. So just give me a minute.
Histidine kinase like protein.
Histine kinase like protein. Just a minute. Okay. Histine kinase like protein. Yeah, fine. Histine kinase like protein. I will be adding it. Answer, no idea. If you know it, let me know.
Histidine, sir.
Are you sure? Yeah, if that is the,
yes, sir.
yeah, sure. See, if you give me, maybe since you are like like you want the class that happen immediately, that's why I have not referred many things. So if might have given like 1 or 2 days extra, I might have referred other kinds of things. I might have given the answers also, but still no problem. After 2 days, I'll be making another video manually without you. That there I will give you the answer. Okay. So anyway, maybe histine kinase is like protein. Please refer once again. Okay. The next one, which of the following characters is not true about the virusoid? So this is also there, but light doubted about the options which was given. Like they have given options like uh, 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 encapsulation dependent on plant virus. Kailas, I can hear you nothing from you, but because your voice is completely like.
So virus dependent replication, virus independent replication, like that option was there.
Okay, just give me a minute. Options: virus dependent replication, virus independent replication. Okay, virus independent replications are there, right? Okay, fine. So this is also leaving up to you only, because again slightly viruside what we might have studied, we need another helper virus for encapsulation, all the other kinds of things we have studied, right? The next one, this is also a direct question. Shape of the disease progress curve in polycyclic diseases. This is a direct question. What is the answer? It is sigmoid only. This is a direct question. We have discussed this one. Again, the next one, choose the correct statement regarding the satellite viruses and satellite RNA. Satellite viruses can encapsulate without helper virus. Again, this was given. Like the only thing is whether they have given the correct statement or like to identify the wrong statement. That is another problem. That's why I have not given. If they have mentioned the correct statement, this is the correct statement. Like satellite virus is dependent on helper virus for the replication and encapsulation. That is the correct statement. Which means other things also they have given. Like encapsulate without helper virus, that is also wrong one. Replicate independent of helper, that is also wrong one. So if I am right, like if they have mentioned only choose the right statement, the answer is this one. Satellite viruses are dependent on helper virus for replication and encapsulation. That is the option. But I was a little bit doubted about whether they have asked for the right statement or the wrong statement, other kinds of things. Okay, please look into that one also. And another thing, okra initiation leaf curl virus is.
So this is right. 58 question was right.
Right only.
Like
then no problem. So the answer is D only. Okay. Then 59, okra initiation leaf curl virus is transmitted by. What it is transmitted by? Bemisia tabaci. Mostly okra, yeah, mostly Bemisia tabaci, white fly. Mostly this okra bend related things are associated with this Bemisia tabaci. Okay. The next one, which of the wrong statement regarding the spread of the diseases? Wind, aerochory, water, hydrochory, animal, zoochory, human, anthropochory. What is the wrong statement?
A will be the answer, but in that question, so it was like A, B, C, D, and all like that. And they have asked like.
See.
Like statement wise questions, sir.
Like just a minute. 60
like
statement.
They have put the options like B, C, D correct, and all are correct.
See, so they have here it is
yes,
given
sir,
is
all was correct.
Here they are given is identify the wrong statement along with statement wise. A, B, C, right, all are right. So what is the answer?
Answer B, C, D, and right. Wind is anemochory.
See, aerochory is otherwise called as anemochory. Aerochory is otherwise called as anemochory. That's right. If you want, you just go and type in the internet anemochory. Yeah. That is the problem. See how they actually somewhere they have twisted. See, if you read 2 to 3 times, you will get it, because by seeing it immediately aerochory, we may get it confused. But anemochory is otherwise called as aerochory. You'll just type in internet, you will get it. Aerochory is also, you can also call a synonym of anemochory. Okay. So I believe all the options are right. If that was there as a statement like A, B, C, A, C, the answer is all the above. Okay. See, don't worry. Yeah, by discussing the things, you may thought that yeah, I made a lot of mistakes. Of course, I also made a lot of mistakes, man. There is no point of looking back, because you cannot change the past now, right? There is also a couple of memes for you guys at the end, for fun. Okay. Anyway, 61. Which fungal pathogen is responsible for chytridiomycosis in amphibians in a severe, often fatal skin disease characterized by hyperkeratosis and disruption of the normal skin? It is actually Brachychytridium dendrobetalis. I am really thankful for my mycology videos. I still remember this one. In one of the mycology video, I made this one. So hyperkeratosis in amphibians, especially frogs. It is related to Brachychytridium dendro. Yeah, please.
Sir, in that one option was in Chytridium endobiotic was also.
Chytridium endobiotic. Okay, just give me a minute. One option, Synchytridium endobiotic. Yeah, that's right. If I remember, that is the option number D, right? Synchytridium.
I don't know, but it was there.
Exactly. Endobiotic. I think they only twist the questions, shuffle the questions. Options remain same, I believe. But anyway, no problem. I will change that option. 61 single remedy. Anyway, next one. Ponticarvo Roper discovered the parasexuality in 1950. Which of the following fungi? Here you need to think very carefully. Option: Penicillium rhizophus, Rhizophus mucor, Aspergillus penicillium, Aspergillus rhizophus. Which one is the answer? Aspergillus penicillium, because why? Rhizopus is already established. Yes, Rhizopus already established sexual reproduction is there. Again, mucor is also same. Again, Rhizopus is also same. There is only one option they have left: Aspergillus and penicillium. So originally it is discovered in Aspergillus. Later, I feel probably in the year of 1958 and 15, they also discovered the same Ponticarvo discovered in penicillium also. Probably 1959 or maybe probably 1960, close to, in penicillium. But originally this phenomenon 1952 only, Aspergillus and penicillium. Okay, next one. Choose the correct statement regarding the bacterial endospore. Like options will be like endospore coat contains 80% proteins, endospore resistant to heat due to black thick wall, or like heat due to thick wall. There is one option C, and the last option I remember all the above. But there is one option C I didn't remember properly. If you know, please let me know. 63.
It was the name of protein, I think. The endospore have some protein.
Dipicolinic acid was not there, if I remember right. Name of protein, some protein was name was there, but I didn't remember properly.
Sir, actually it was like endospore has same characters like spore.
Yes, I think that option was there. Right. That
Yeah,
option
some
was there.
typical option was there. Some
Yeah.
CD.
C, endospore.
Sir, actually it was statement type question.
Yeah,
Means A,
spore.
B, A, B, C, like that was there.
A and B correct, C and D.
Answer I wrote A and B was correct.
So only A is correct, sir. No, only A is correct.
A, B, C. Yeah, that kind of thing was given. So this is one of the question that was asked, right?
What is the answer, sir?
See, endospore contains 80% protein. That is a wrong thing. See, no. It contains endospore resistant.
Only A is
Wait. Endospore resistant to heat due to thick wall. Of course, it contains thick wall. It is heat resistant just because of consist of DP and calcium ions. Probably B is wrong. Maybe C, if you whatever you are saying, like endospore is as like if a normal spore. Yeah.
So in this type of question, 2 were correct and 2 were wrong, like that option was there.
Yeah. If you remember properly, please let me know. Okay. Just give me 1 minute. ஹலோ, நான் கிளாஸில் இருக்க முடிச்சுட்டு பண்ணுவா. ஓகே, லேட் ஆயிரும். Yeah, so this things was there. Like if the C option, like endospore is like a normal spore, if it is mentioned, the option A only, right? Okay, so please recall the options. Please let me know. Yeah, tell.
No, sir. Screen was not visible. That
Now it is.
now it is visible.
Now it is. Okay, fine. Okay, fine.
Yes, sir.
Yeah, fine. We'll finish it a little bit fast. Okay, next one. Example of autoecious macrocyclics. Among the option it is given is Puccinia helianth is the right one. Okay, the next one, genetic material. This is a direct question. Genetic material of rice tumor virus. You know it, right? There are two viruses: are rice tumor spherical form virus, rice tumor bacilliform virus. Whether we have discussed this one, including the genetic material, yes or not?
Yes, sir.
This we have discussed in our classes. If you want, I will show you also. There is one slide, especially in the. Take a look at this one. There is a one slide. We have discussed this earlier. See, RTSV single stranded DNA, RTBV single stranded RNA, RTSV double stranded DNA. It is RTB, which means we have discussed this. They have given an indirect one, which consists of only single stranded DNA, double stranded DNA, single stranded RNA, and double stranded DNA. Like that, it is option C. Okay, it consists of both single stranded RNA and double stranded DNA. Okay, the next one, most commonly used indicator plant viruses for the spread studies or something like that they have given. So the answer is actually Chinapodium amaranthine. I think they have given other things like nicotinamide bentham, nicotin tabacum, claxylem, something like other things will be given. But it is option actually Chinapodium amaranthine. Next one, which of the following fungicide is not a biocide? It is mentioned definitely it is fungicide, not a biocide. Which one is companion, aspire, glyogod, rootstop? What is the answer?
I think it was like a fung.
The biocide. They have mentioned biocide. Which of the following is not a biocide? Sorry, come again. What is the answer? Yeah.
Companion,
Yeah, among the given option, companion is not a biocide, because actually companion, I think it is related to bacillus related thing, bacillus subtilis or something like that. Actually, it is one of the trade name for this bacillus related thing. I think that was actually bacterial related thing, because the remaining option all are related to fungi only or biocides only. Okay, probably it is companion. Okay. The next one, black heart of potato is due to, this is also one of the direct thing, but they have given a long lengthy one. Black heart of potato, it is due to low oxygen and low aeration, I think. The next one, development of initial black and dark brown necrotic lesions at the center of the tuber usually occurs in the potato exposed to poor aeration store. So many stories are there. They give black tip of mango, citrus follicle, black heart of potato, pala blood sugar. What it is?
Black heart of potato.
It is black heart of potato. You can remember this black, like you can sort it out this question by using only one. See here, poor aeration and stored at excessively high temperature. This is an indication that black heart of potato, like poor aeration is there, which means less oxygen and high temperature. So that is the prime reason for this black heart of potato. So black heart of potato. Another thing, application of boron helps to reduce. See, these three things are the direct 68, 69, 70. These three things we have discussed. Yes or not? But in different way, but this we have discussed. Application of boron, general black tip of mango is due to boron deficiency and also sulfur dioxide concentration they have given. So boron, application of boron helps to reduce the severity of whiptail of cauliflower, black tip of mango, citrus foliocelis, mango anthraconas. I am telling you once again, one of the two options were we have added for our convenience, but what is the answer here? Boron deficiency. So if you apply, what can we rectify? Yeah, so back tip of mango. Now if you calculate, definitely there are at least 70-75 questions you can easily answer it. The only problem is you guys are like feared a lot, right? There are questions, 75 easy questions, you can easily answer it without any negative marking, you can easily answer it. But anyway, no problem. Let's see. Next one is manganese deficiency causes, which means which of the following is a manganese deficiency? Gray speck of oats, white bud of, yeah, mango. It is a gray speck of oats. This is also a direct question, right? This is also we have discussed. If you want, you will just go redirect back to this physiological disorders. There we have discussed this one. I have given this one, right? This is also direct thing. This is one of the UG thing. Number of rings present in gram negative. You know it. Positive 2, gram negative 4, LPMS. This is also direct question. So the answer is B. Okay. The next one, which curve are used for the differentiate epidemic rate of two different diseases? So
No, sir, they mentioned the speed. They mentioned the speed.
wait. 73. Tell now.
They mentioned the speed of the two diseases like that.
Speed of.
Yes, sir. It was speed related to something. Spread or speed.
Comparison, sir. It was like comparing. And the question was something like comparing the two diseases.
Please, every three of you. Wait.
Sir, they mentioned comparing the two epidemic speed of the two different diseases.
Kailash, Abhinav, Kirti, this is your work. 73. You make your own versions and send me along with the option. I will finalize one. Okay. The three of
Sir,
you.
let me tell you, sir. Sir, it was given that two epidemic diseases are there, and we have to compare its speed or spread. So, what curve we can conclude?
Yeah, you
that?
make it. You
So
make it and send it to me. Yeah, it is AUDPC, and by seeing it, like if whatever you have mentioned, like the different thing, it is AUDPC. But I feel different rates of the diseases that can be measured by using this disease progress curve also.
Yes, sir. The answer is epidemic rate curve only, because they are asking the speed of the two diseases like that they are asking.
Anyway. You make your own versions and send it to me. Probably it is AUDPC or maybe disease progress curve. I feel it is AUDPC. Okay. You make your own version, send it to me. I will refer and let you know. Okay. The next one, indicator plant used for citrus schizovirus virus. This we have discussed 1000 times. What is the answer? See, we have no time. Please tell me. Yeah, Claxylem you can call it as, yeah, you can call it as Mexican lime or Mexican nimbu, something like that in Assamese. Okay, his answer is Claxylem. This is also direct question. So this is also a direct question, like association between two different organisms where one get benefited and another get no harm, no benefit, nothing. What it is called? Mutualism, amensalism, commensalism, parasitism. What it is? It is a commensalism. It is a direct question. See,
Actually, amenasalism was not there enough.
Instead of that, what was there?
I don't know, but amenasalism was not.
Okay, no problem. No amensalism was there. I will sort it out what exactly it is. Okay. Anyway, my answer is commensalism. See, by seeing this, can you tell me the question paper is difficult now? But long descriptive was there. Can you tell me the question was difficult? Of course it
So
is not.
sir, pressure was there,
I
and
I
lots of
understood. That's why. Yeah,
long
that's why.
description question was
That's
mixed
I
along with
understood.
these
I
questions.
understood.
So
That's why. That's what I'm telling. I totally understood. That's what I'm telling. See, there are. I just calculated. There are 70 to 80 questions you guys can answer in your fingertips, just because of the pressure and long descriptive things. Maybe like you just maybe committed the other wrong things, other probably, but still it is an experience.
Sir, sir, the main problem was with the mouse and the screen.
See, this is the problem with ASRB. This we can raise it, because if that is the problem, we can raise it, because it is the duty of NTA, National Testing Agency, to take care of everything. If they cannot arrange all these things, that again this is a problem. See, we will sort it out everything. Please stay until the, even though it is taking a little bit half an hour extra, please stay until the last slide. We will discuss, and I am going to write all the problems. We will make a mail with all your approval. You will, every single of every one of us, we will send the same mail to ASRB. Let's see what kind of responses we'll get. Okay, because only one will send, ASRB won't respond, trust me, because it is not like a normal thing, right? It is one of the prestigious things, right? So if the many people will raise, definitely we'll get a maybe some kind of responses, other things. Okay, the next one, sugarcane set treatment temperature for the disease control of sugarcane ratoon stunting. 50 degrees centigrade, 2 hours, 2.5 hours, 3 hours, 3.5 hours. What is the answer? Here two problems are there. Yeah, answer is 50 degrees Celsius for 3 hours or 50 degrees Celsius for 2 hours. Two things will be there. In one of the thing, directly that was mentioned by the scientist from probably.
when it says 2 hours.
Yeah, so 50 degrees Celsius for 2 hours, in one article it is there. You will just I will show you. You will just type the same question. You will get 2 hours, 3 hours, 2 hours, 3 hours. Both you will get. That is the confusion arises. I have gone with option A because in my book also I have mentioned 50 degrees Celsius for 2 hours. I have gone with option A. That doesn't mean it is true. In the articles, it is also mentioned that 50 degrees Celsius for 3 hours. There the confusion arises. Because I have mentioned A, that doesn't mean A is right. That is also maybe wrong. That's what I am telling. That's why I have not given the answer. Maybe A or maybe C. Probably C is the right thing, if I believe. Okay. So next 77. Black cut of potato is endemic in which of the following hills? This is not a direct question, but still answer there. Aravali Hills, Nilgiri Hills, Common Hills, and Karakoram Hills. Common Hills. Oh, sorry, Coman Hills, Karakoram Hills. Black water potato. Maybe you studied Darjeeling and all those things. Don't worry, because Darjeeling was not there. That doesn't mean options was not right. What is the answer?
Nilgiri.
No, it is not Nilgiri. It is a Coman Hills, because this Kumal Hills, if I am right, it is there in Uttarakhand. It is mentioned. Yeah, this is the problem. Okay, in Kuman Hills, I read this in Kufri, one of the CPRI published one of the thing like a bulletin. I studied very long back, but somehow I never attended this question. By I thought that maybe Darjeeling might be there. Later I referred it is actually Kuman Hills. And it is the black cut of potato infection is also reported. It is an endemic in Kuman Hills also. Probably the answer is C, but I don't know whether this ASRB will consider such kind of question. That's why I told you in the group, please make a list of questions, whatever that we need to ask. I know that none of you have made it, right? Is I am right? None of you had made it, right?
I wrote that on the
Yeah. So it is not actually. Yeah. We will sort it out. This kind of questions, we will sort it out and we'll just request. Okay. The next one, partial root parasite that can grow in the absence of host. Which one? Cuscuta, Loranthus, Warbank, striga. This is also a direct question. Which of the following? It is actually striga. Yes, striga can germinate without absence of host. Without host also it will grow. Okay, next one. A single flower of striga produces how many seeds? Because everyone got confused. Striga plant produces, but they have mentioned very tricky thing. A single flower. They have mentioned this point. That's why I read question 2 times. Why they have given this kind of options? Because we never read such kind of things. Because a single striga plant produces an average of 50,000 or a maximum of 5 lakhs. Sometimes we go up to 9 lakhs also. But why they have mentioned? But I read once again. There I realized they have asked a single flower. We never expected this kind of things. But what is the answer is, again it is a doubt, because in one article it is mentioned as 1000 to 1200. In one of the Springer article it is mentioned as 12 to 1500. In one of the article it is mentioned as 15 to 1800. If I asked the same thing to Gemini and the another one chat GPT related thing, they have given 15 to 1800. But if I asked the resources, where exactly they have given different resources, each resource they have mentioned different thing. But I sorted out, like the majority of the sources they have mentioned is 12 to 1500 and 1000 to 1200, like that they have mentioned. Maybe 12 to 1500, or maybe D. I have no idea. If you have ever encountered a single flower somewhere, if you have seen somewhere in the articles, please let me know. Okay. So the next one, which of the following is a facultative hemiroot parasite that infect crop plants? Observe very carefully, which of the following is a facultative hemiroot parasite? Since it is mentioned hemiroot parasite, directly will go to Warbank, right? Here they have mentioned is facultative hemiroot, probably Rhinanthus, might be Rhinanthus. I think it is also given in my book also, probably Rhinanthus. Please recheck once again, because the since it is mentioned as facultative hemiroot parasite, might be Rhinanthus. Okay. The another thing they have mentioned, like methods of recombination in bacteria, but I remember the right sequence, like conjugation, transformation, transduction. This is the method of recombination. I think they have also mentioned A, B, C. Please, if you remember, please let me know, or else please send me separately. Okay. The next one, which of the following plant pathogen? Are you guys are there?
Yes, sir.
Okay,
Yes,
please.
sir.
Yeah, please. We'll finish it. We'll finish just 5 minutes. 10 minutes. We'll finish it. Okay. Another thing, like which of the bacterial genera they produce monotrichous flagellation, strictly aerobic, gram negative, muciloid, biochemical reaction, gram positive, oxidase, catalase. Yes. How you can recognize this is a Xanthomonas? One point is there.
Yellow pigment.
Yes, they have mentioned two things. One is the yellow pigment and the xanthomonidae. So by seeing the two things, you can come to a conclusion. This is a Xanthomonas. So the answer is Xanthomonas. No further confusion.
Actually, instead of Pseudomonas, they mention Corynebacteria.
Instead of Pseudomonas, they have mentioned Corynebacteria. Fine, no problem. Corynebacteria. Okay, I'll change it. So I also request you, if you also contribute from your side by changing the option, other things, it will be easy for me. Okay. So another thing, identify the wrong fungi and toxin pair: ten toxin, Victorian, fasciolotoxin, tap toxin. It is a tap toxin. Tap toxin never produces alternative solvent, right? So the wrong pair is tap toxin. Okay. The next one, what is meant by pathovar? A bacterial strain. So many things will be there. So options also I have not remembered properly, but whatever I have remembered I have given. But the answer is this one only. A bacterial strain or set of strain distinguished by their a specific host plant they infect. So that is generally it's a pathovar. Other kinds of options which were there, it is not properly remembered. But if you know, please let me just take a screenshot and send me if you remember. Okay. Next one, tulip color breaking belonging to the family. This is also one of the question. If I remember, yeah, Potivirus. If I remember, this is also one of our model question paper question. It is actually belonging to Potivirus. Okay. Next one, hypersensitive reaction is a layer of defense mechanism applied to plants: susceptible plant, resistant plant, moderate susceptible, moderate resistant. What it is?
Resistant plant.
It is actually resistant plants. Okay. The next one,
Resistant.
Poca Boeing disease. This is one of the model question paper I have given. Two questions was there. One is Poca Boeing disease caused by. Second one is Poca Boeing disease was first discovered by Went and some other scientist was there in 1896, if I remember. What is this species associated with Poca Boeing disease of sugarcane? It is Fusarium. It is Fusarium, no doubt. There are multiple species of Fusarium which is responsible. It is Poca Boeing disease caused by Fusarium. If I remembered, we have discussed the same thing. Like there are few stages, are there knife cut stage or something like that we have discussed. It is actually caused by Fusarium species. Okay. The next one, angular leaf spot of cucurbits. I repeat, it is not a cotton. Angular leaf spot of cucurbits. So direct question, which is caused by Pseudomonas syringa pathovar lacrimis. Very, 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 very important. Even for UG, even our teaching UG also we used to study this one, right? So cucurbits angular leaf spot, it is a Pseudomonas syringa pathovar lacrimis. Okay. The next one, organization governs nomenclature of viruses. It is ICTV. I think their options are a little bit different, but the option ICTV was there, like SAC, ISM, ICCC, IPC, something was there. But if you remember option, please send me. But the answer is ICTV. I think everyone might have answered this question, because there is only one organization. There is only one organization.
Sir, one question was, sir, I don't know if you have mentioned, like one question they have asked, they have written 2 or 3 lines. At last, they have written cellular defense structure. What are the TR proteins and hypersensitive reaction.
Yeah, that was there in the upcoming. Okay, that was there in the upcoming. We'll see it. Okay, see, I'll be sending this copy to everyone. Please go through once again. If there is any corrections, please let me know. I will change from my side. I will make it as a full video in other way. Okay, by identifying the answers also. Okay. Next one, scientists who made contributions in the morphology and observations of plant viruses. Bijernek, Stanley, Krause, Bowden and Pyrie. See, absolutely no Bijernek, no Stanley. Here the confusion arises, whether it is a Krause or Bowden. So maybe maybe the chances of Krause is more because they have given the morphology of the particle of virus, something like that they have given. Because see, Bowden and Pyrie is also related to this.
They mentioned only the virus, morphology of virus, detailed study of morphology of virus.
Detailed study of morphology of virus, something like that they mentioned. Probably Krause or maybe the chances of Bowden and Pyrie, because the nucleic acid and protein, that I mean like nucleoprotein. So I was a little bit confusing. Probably it is Krause et al. Because they have mentioned the microscopic, who initiated the microscopic studies of the morphology, right? If I am right, it is mentioned as microscopic.
No, sir. They mentioned detailed study of morphology of virus. Right?
Yeah, maybe. So probably Krause, maybe Bowden and Pyrie also, maybe Krause. So just take a look into this one. Yeah, next one. Numbers of clades of Mycota. Yeah, this is a direct question. Thanks for my mycology videos. At least I remembered this one. Actually, in Mycota, there are 9 clades are there. So the answer is 9. Because since I was doing right now, I was doing the collecting or gathering all this information. That's why I remembered this one. Actually, in the Mycota, 9 clades are there. Okay. The next one, trips related to. First of all, trips means you know it, trade related intellectual property rights. It is purely related to World Trade Organization. None of the organization is involved. WHO, IPPC, FAO, this is not related. It is purely related to WTO. I think every one of you might have answered this one. So the next one, which of fall?
No, sir, I didn't answer.
Yeah, that's okay, no problem. Generally, it is the trips, other related things is actually related to WTO only, World Trade Organization. See, this question paper, you take it as an experience. I took this
Sir,
question.
I just guess about that WTO, but I didn't repeat this question.
Yeah. See, take this question paper as an experience. I took this question paper is like a slap, because hereafter the way of preparation and the model, everything will changes. So everything, whatever happening, take it as an experience. Everything happens for our sake only. Okay. So anyway, next one, following is not true about the viral. Following is true about the viral management. See, planting resistant varieties, seed health, spraying the insecticide to manage vectors, cultural practice, all the above. Maybe all the above. Probably, I'm not sure, because all the options, there is nothing wrong with the options, 3 options that is given, because for the managing up.
Actually, in this border crop was also there.
Border crop maybe somewhere might be there. Probably, see, there is no wrong with seeing these 3 options, right? So maybe option answer might be all the above.
Statement answer was there.
Yeah, statement
1, 2, 3,
kind.
1, 2, like that.
Wait, 93 statement kind. Okay. Okay. So this was probably all the above. I'm not sure. I'm telling once again, probably all the above, because I have gone with all the above. Because if you see these 3 options, there is nothing wrong with these 3 options. That's why might be. I'm telling once again, might be. Okay. So which of the following term is related to sudden and severe spread of a disease in a large area in a short span of time? This is also a direct question. What is the answer?
Epidemic.
It is epidemic only. It is epidemic. Within a short time, it spreads a long area. It is an epidemic. Okay. Please stay until the last slide. I just want to tell you one more thing. Okay. Next one, which of the following is wrongly matched? Epidemic, endemic, pandemic, sporadic. What is the answer? Epidemic means spread of the disease, large geographical area. Disease confined to a particular area. Pandemic spread across the country, everywhere. Sporadic, disease occurs in an area regularly in a severe form. Yeah, sporadic is the wrong. Yeah, sporadic, because it occurs actually irregularly. Sometimes maybe severe, sometimes no severe. Not very severe. Sometimes very rare cases you see severe. Most of the times no severe, like Udbatta. Okay. So answer is sporadic. Maybe it is also kind of a statement kind of things they have given. Anyway, no problem, leave it. Okay. Next one, induced systemic resistance is operated by PR proteins, phytolactin. There is one question they have given in this way, but I actually not remembered this question. If you know, please let me know. I'll be sending this soft copy. Please make a changes. You just tell me question number and what are all the changes you have observed. You just tell me that one, because if you made changes, again it will be difficult, because every one of you made a lot of changes. You just tell me that like question number, and what are all the things that need to be changed, I'll be very happy. I will change it. I will make it another. Okay. The next one, 97. Which of the following is not a signaling compound of induced resistance? Salicylic acid, jasmonic acid, ATPase, and fatty acid. What is the answer?
So fatty acids.
No, that's not true, because fatty acids are the alternate derivatives of this signaling compounds. Might be ATPase. Yeah, might be ATPase, because I also wrongly mentioned this fatty acids only. But coming back and realized something, and I referred, I came to know actually this fatty acids actually derivatives of this signaling only. So probably this fatty acids also a part of this signal. This ATPase I later realized, because the ATPase what it will do, because it will convert from ADP to ATP, like just conversions it will do. So maybe I repeat, maybe ATPase. Please remember the disclaimer than the first slide. Maybe ATPase. I'm not sure. Maybe fatty acids also, because I have gone with fatty acid, but later I realized probably the answer might be ATPase. Okay. Because one point I'm sure this fatty acid is also one of the derivative of this signaling. Okay. Next one, injectosomes. I am not sure whether they have mentioned this line injectosomes, but I am sure they have mentioned bacterial effectors. They have injected into the cytoplasm. So, but one point is very clear.
So question was like it is directly transferred to the bacterial host cell.
Wait.
Injectosome was not there.
Yes, that's what I'm telling. Directly transferred, right? Directly transferred to bacterial cell. Okay, fine. So, what is the answer? It is a direct question. Still, it is a direct question only.
Type 3.
Type 3.
So type 3.
Yes, there is no doubt. It is a type 3. I think injectosomes was not there. I will revise it. Okay. So definitely it is a direct question. It is a type 3. Okay. Next one, anthracnose of grapes is epidemic in France and a serious disease in many countries, including India, introduced in there. I'm not sure. Probably 1950. I'm not sure, because I have not attended this question, and I tried a lot to find the answer also, but I have not get it. If you know somewhere else, if you see somewhere else, please let me. Next one, bentonite clay is used for the virus transmission studies due to uniform spread of the sap over the plant disease, protection of virus particles during the mechanical transmission, it able to dissolve the virus proteins, maintains the pH. What is the answer? Your voice is not clear. What is the answer?
Uniform spread, sir.
Yeah. See, here two problems are there. Option A and B both are right only. It is used for uniform spread and also used to protect the virus particle during the mechanical transmission. If we need to select only one option, the B will be the right option. Of course, the A is also right. I'm telling you once again, A is also right. It is used for uniform spread. It is also helpful to protect the virus particles during the mechanical transmission. So,
But I have selected B option.
yeah, whatever it is, I'm just telling both A and B only right. If B is right,
I think bentonite clay. Sir, I think bentonite clay is the example of good spreader, no, sir.
yeah, that's what I'm telling. See, I have also gone with option A only. I'm telling you once again, there is a problem, because both A and B are right. Uniform spread, it is also helpful for uniform spread, like overall spread, uniform spread. The second one is it also helps to protect the virus particles also. So which one you go with, it is up to you only, because I am just telling the, rather than what I have attended, I am just telling you what is the probabilities. I have also gone with option A only, uniform spread of the sap over the plant surface, because I read it, because we in UG and PGs we might have done this one, like sap transmission experiment, right? So I have gone with A, but option B is also the right answer. But if I want to go with only one, maybe B is the right one. I'm not telling, just it is an like just a normal like assumption only. Okay. Next one, wheat stem rest is a dangerous macrocyclic. See, forget about all these things. See, which of the following spore is a resting spore? Teltospore, bestiospore, iridospore, aesiospore. Direct question. Which of the following? Stiltospore. Yes, iridospore is a like non-resting spore. Yeah, repeating spore. You can also call as repeating spore. That's right. Teltospore, resting spore. Answer, direct question. Teltospore. Now tell me whether the question will be difficult, which means long thing will be there. But still, I would say 70-80 range. Okay, fine. Next one, Mungbin yellow. This is also direct question. Gemini viridae, right? Mungbin yellow mosaic virus belong to Gemini viridae only. You don't worry. I will making another thing. I'll make the things very easy, because I'm making another thing, like how to make this virology thing very easily. I have a very big tree of like making all these things in easy way. Once it is done, whether you are a member or not member, I will be sending you. Don't worry about it. But it will take time. Okay. So Mungbin yellow mosaic virus originally it is Gemini viridae. Okay. Next one, fungal mycelium consists of a specialized septa for special functions to be done. It is also direct question. Special, complex, bordered pit, simple septa, developers. What is the answer?
Dolipoceptor.
It is Dolipocepta. It is also direct.
Dolipocepta.
Yeah, it is Dolipocepta. Okay. See, this is also we have discussed, right? Bacteria consist of a single flagellum on either side of the bacteria. What is the answer? Amphitrachus. You know it. This is a flagella either side, right? We have discussed atrachus, monotrachus, amphitrachus. This is discussed it, right? So this is amphitrachus. Direct question. Next one. Which of the following is used a vector in transferring of genome by genetic engineering? What is the answer? Plasmid, bacteriophage, plasmid, bacteriophage, delo-hybrid.
Plasmid and bacteriophage.
Yes,
C.
both option C, plasmid and bacteriophage, because in bacteriophages we studied two things, lytic and lysogenic phase. The lysogenic phase can be exploited in transmitting or genetic engineering purposes. So the answer is both, like plasmid and bacteriophage. Okay. Next one, vericolvin A inhibits the germination of. I'm not sure whether it inhibits or whether it activates the germination. Whether it, what I have mentioned, whether they mentioned inhibits or what.
Yes, sir, inhibit only.
Inhibit only. Okay, but maybe I'm not sure. Probably.
The answer is Favabin only.
See, I have gone with Favabin only, but I thought maybe Vorabank is also a chance, because I read somewhere this Vorabanki is also where this compound was produced, like Vericolvin. I was doubted whether it actually enhances the germination or decreases the germination. I have doubted about. See, for I also gone with Favabin only, because I have my own reasons by seeing this question. But I was a little bit doubted there is a chance of Vorabanki also. Please refer once again. So if you like, if you give me 1 or 2 days, I will refer and let you know. Okay, I was actually working on this one. Okay. Next one, on the basis of pathothallus. I'm not sure whether they have mentioned this pathothallus or what or not, but this was mentioned, the region where EPS production is more. Rhizosphere, phylloplane, rhizoplane. What is the answer? It is actually rhizosphere, where this rhizosphere region only where this maximum production of EPS will be there. Okay. The next one, answer is rhizosphere. There is no doubt. There is no doubt. I referred this book. Okay. So next one, which technique is used for the detection of plant viruses? PCR, serology, electron microscopy, all the above. Maybe all the above, I'm not sure, because this PCR, serology, electron, all these used for the identification or detection of plant viruses, right? So maybe all the above, may not be also. Next one, magnification of virus particles of EM is based on. I completely no idea about what exactly they are asking, especially in the question number 109. Magnification of virus particles is EMV based on what? Magnification 2 lakhs 100, 1 lakh 1 lakh 50,000, 200, 1 lakh 100, 2 lakh 200. I don't know what it is. Maybe based on my just searching or somewhere I found this 1 lakh to 100, but I'm not sure. I'm telling you once again, I'm not sure. Next one, pH of a medium can be checked by. This is a basic question. Tell me, before autoclaving, after autoclaving, after cooling, after autoclaving and cooling. What is the answer? pH is always checked before or after. Come on, man. pH can be always checked before autoclaving, not after autoclaving. pH is always checked before autoclaving. When we are, like when we are making this medium, once it is done, we will check the pH and then we will do it, right? If there is an adjustment need to be done, we will do it and then we will autoclave it. So the answer is before autoclaving. Are you guys are there? Finish little bit faster, okay? So this is one question regarding the magnification of particles of EM. Like I didn't understood what exactly the basis for this. I'll just look into this one. Next one is the pH of a medium can be checked by. What is the answer? Before autoclaving or after autoclaving or after cooling, or what it is?
Before autoclaving.
Before autoclaving.
pH of a medium is
I think after autoclaving and cooling.
actually pH is always checked. Yes, no. pH is always checked before autoclaving. And once we prepared something like a mid any kind of thing, we'll check the pH and we'll go for further. Okay. So if there is any corrections or further things will be there, it will always done before autoclaving only, which means so the answer is A, before autoclaving, not after autoclaving and cooling. It is actually before autoclaving. Okay. Next one, function of PR protein. They have given
Sir, what
something.
is the answer for conjugate? What is the answer 109? Sir, what is the option for 109?
Wait.
So from 107, you were not audible.
Oh, from 107 onwards, I'm not visible. Okay, fine. See,
Yes,
so
sir.
the okay, fine, no problem. The maximum EPS production will be there in the rhizosphere. Next one is which of the following technique used for the detection of plant virus? They are given PCR, serology, electron microscopy.
Actually, instead of rhizoplane, the option was endosphere.
Instead, Wait. Yeah, I think
Sir,
one
my
option.
sperm was also there and endosphere was also there.
Wait. Actually forgot. Last
So
one.
pain was not there.
But anyway, please, anyway, no problem. I'll just change the options, or like you just tell me the question number along with mention the option. I will change it because somewhere I have pen. Okay. What is the option? Tell now. Spermosphere, that was there. Another one,
Spermosphere as endosphere.
androsphere, that was there, but the answer is rhizosphere only. Okay. The next one, which of the following is used for the detection of plant viruses? Maybe all the above. I'm not sure. Maybe all the above, because PCR is used, serology is generally used, electron microscopy, all these are used for the detection of plant viruses. Maybe all the above. I'm not sure. Next one,
Yes, sir,
magnification.
all the above.
Maybe that's what I'm like. Maybe. Okay. Next one, magnification of virus particles is EM is based on. I didn't understand what exactly the base of this question, but I have gone with option C for some reason. I have also searched. They have given different options.
Even tomato.
Yeah. And one more point, like
Sir, the basis of them giving this 100 particles is not justified, so I typed that question.
yeah, that's what I'm telling. So what exactly the base, in which way they have considered, I'm not sure. See, I have gone with option C. That doesn't mean option C is right. I'm telling you once again, maybe option C or may not be also. So this is one thing. I still don't understand what exactly the basis of this question, what exactly this is made. But anyway, no problem. Next one, pH of the medium, it is always before autoclaving only, no doubt. Okay, it is 100% sure, because even in our UG, when the experiments is carrying, we always check the pH before autoclaving only. Okay. Next one, functions of PR protein. I think there is a slight variation in the questions also. Cross linkage of the cell wall, inhibit the spore germination, plant defense or mechanism, all the above, something will be there. I'm not sure about this one, because the question itself is a little bit confusing, and options also slightly various. Okay. Next one, polyethetic or polythetic classification is related to, what is the option? Fungi.
Hello.
Yeah, tell please.
Sir. Sir, is PR protein is inhibiting the spore germination? I have that doubt.
Of course it will do. See, the PR is the ultimate
Yes,
level.
it can do.
Of course it do. It is the ultimate level. So there is nothing can penetrate this through this PR protein. Once it produces, that's it. That is the end of the pathogen. It will do. Okay. But there is a, like, the question they have asked in different way. If I am right, I'm not remember the question properly. If you remember, or
No,
any one of
they asked in a statement. They
the,
asked statement 1B, 1 and 2 is correct, 2 and 3 is correct, all are correct, none is correct. Right?
yeah, that's why something like that they have asked. But if you remember, please let me know. But they have given in this way. Okay. So the next one, polythetic classification, is related to the classification of what? Fungi, virus, subviral, bacteria. What it is?
Subviral.
It is actually no. It is actually virus related thing. It is actually, because in the ICTV classification, this polythetic is one of the group where this virus related things was classified. So if you have ever come across the answer of option of subviral agents, please send me back once again, because I have referred this couple of articles in the ICTVs also, but they have mentioned it is related to virus. But in some case, it is also referred for fungi only, but it is primarily associated with the virus only. Because if you have, if there is any reference related to subviral, please send me back. I'll refer also. Okay. The next one, which prominent, just 5 more questions if I am right. Which prominent genus of gram positive bacteria is distinguished by its characters of branching filamentous? See, no need to study long. There is a one point that, yes, streptomyces. There is a one point, filamentous. That simple point that will give you, this is a streptomyces. Okay. Next one, which are the following statement? I'm not studying. Statement 1, plant disease caused by plant disease epidemics and control describes the principle governing the rise and fall of the disease in a plant population. Second one, epidemiology definition, something like that, but not mentioned in Van der Planck book, that study and spread of the something, it will be there, but it's mentioned both statement A and B correct, A and B is not correct, A is B, like Van der Planck given house, something like that was given. It's completely confusing, so I have not left it. I'm left it free. I'm not sure. Please, you just go through and refer it if you find. Let me know. Next one, in disease triangle, which are the factors of pathogen affecting the disease development? Again, there the question is incomplete. There is something else is there in the question. I think they have given a long thing.
Sir, like that was there. The amount of inoculum near to host, age of the host, then level of virulence, or both level of virulence and amount of inoculum near host. Like that.
Yes, sir, it was like that. Level of virulence, amount of inoculum, and Age of I don't think age of host was there.
That is okay, but the thing
So
is,
like that was there.
wait,
Level of
wait, wait, wait.
virulence,
Wait,
amount of
options
inoculum
okay.
near to host.
You forgot about options. Options okay, but the thing is, like the question, there is a like they have given in a lengthy way. I feel they have given a lengthy way in disease development. Which factor of the pathogen
Yes,
affect
sir,
the
they are
development?
asking which factor is which factor of pathogen is favorable for the disease and like that
Yes.
they are
So the level of virulence and amount of inoculum, both. The answer is both. So, but the thing is, in the option, sorry, in the question, I think there is something they have twisted. But I am sure the answer is both A and B. Like the level of virulence and the amount of inoculum near those, both will infect. Like both will play a role. But there is something they have twisted in the question. If you remember, please send me. Please let me know. But
No,
I think
sir, in question they mentioned, sir, in question they mentioned in a disease triangle, host, susceptible host, virulent pathogen, environment, this completes the disease triangle. But among all these things, among the given option, which is a factor which is more close to the pathogen factor, like that they are.
Kailas, I will be sending this copy. You are the in charge. You need to like put me the question number and whatever you remembered, you send me back. I will go back once again and I will confirm it. Now you are the primary in charge. The secondary in charge of the wrong correction, like the other changes need to be done is Abhinav. Okay, you both are the in charges. Go through all the questions once again. I will be sending the copy. You will just don't make any changes. Mention the question number and send me what all the changes need to be done. That I will take care here. Okay. Next one, something like alternating mosaic pattern, something they have given. Another statement kind of deployment gene kind of thing they have given. Statement question, quantitative resistant or adult plant kind of thing, one thing is given. Endophytic fungi, one question they have given. Carla virus, something that is related to Carla virus or Tritimavirus, something like that they have given one question. Isolation procedure of endometic organism, they have given one question. Characteristic of endophytic, that is another question. Pathogen factors affecting the disease development is another question. I think they have discussed this bangle blood need to be changed the question number, right? Question this one, right? Characters of streptomyces bacterium, I think they have asked. Wheat rust genes, something like the rust kind of genes they have asked, something not remembered properly. Favorable conditions for anthrognosis of beans also they have asked one question related to one thing they have asked, like aflatoxin B2 or maybe AFB1, something they have asked. Mosaic of TMV mechanism, something like that they have asked. Another thing is gametangiogomy and somatogamy. There is one thing related to reproduction of this fungi. One question is asked, I didn't remember properly. Another thing is theorem is a
Spermatization was there, sir. Spermatization was there.
That's okay. That's okay. That's what I'm. You are the in charge. You go through all these questions. Yeah. See, I'm sending this soft copy to every one of you. Please go to the question and just mention the question number and tell me whatever you remembered. The rest I will take care and make it into the finest one. See, you have sent me the cut things,
Yes,
but
sir, there was very nice and long question also there for statement based.
that's what I'm telling.
It was related to that, sir,
See
gene for gene hypothesis.
gene for gene hypothesis. Yeah, yeah. That's see. I tried to collect my level best. Just see how many questions I have collected and remembered by single self. If you put your hand also, we can make this question paper into a complete one. That's what I am telling. Okay. So another thing like theorem dithiocarbamates monoalkyl, something like that. There is another statement will be there. This is all about all the question papers. Now, other question number 30, the techniques used for the insertion of desirable genome into the plant, like both a electrophoresis, something like that will be there. Ecology of the fungi indicates, options not remembered, that's why I left it free. Okay. Sexual reproduction method, the one question was there. And again there is a detection method like remote sensing kind of thing, it will be there. So if you remembered all these questions also, just mention the question number and send it me back. I'll be very
Sir,
happy.
in question number 133, sir, in question number 133, there was an option also like their micro injection of plasmid or something, DNA micro injection was there.
No, micro array.
Yeah.
Was there.
Something was there like injecting this kind of thing.
Yeah, that's why you. Yeah, that's why you two
we
are the.
can also consider that option, no, sir?
You something, you both.
Question 133.
Yeah, tell.
Hello.
Tell. I can hear you.
In this option was, post-transcriptional gene silencing, RNAi, as well as mutagenesis, and fourth one is microarray.
That's why you both are the in charges.
That was different question.
Yes, I believe that is a different thing. Anyway, you both are the in charges. You just.
Yes, sir.
Yeah, that is a different one. That is we have already discussed. That is we have already discussed. PTGS, that was all discussed. This is a different one. like for injecting the genome of anything like into the plants, what is the technique? Electrophoresis, like micro arrows, injection, something. There another option, all the above, probably all the above, that is different thing. But the options I have not remembered. That's why both of you giving an option. Please, other members also, please put your hand also. I will be very grateful for your help.
So
because
that injection we can consider now, because in IRI, one of the scientists has also done
not one
this kind of
scientist
thing
here.
by transmitting virus.
You come to South India, I will show you hundreds of
He
people
is
doing in
recently
TNU.
working. I think he's
You
near
you
the
you
lab of Sushil sir. Just to
I
next
know,
to
I
him.
know, I know, I know. See, you come. See, we have published. We have we now we are in the process of publication of one, especially the related to this different methods of insertion that was publishing in Springer. We are in the publication, like in the process. So there I have given in detail about it. That's what I'm telling. See, you come to TNA, will show you gene gun. So all these techniques are actually using insertion of genome. That is a different part. But options I have not remembered. Please, every one of you, put your hand, will make it this happen. Because see, I'm a single individual. See how many I have collected. Like you have gave couple of things, but I still managed to recall the options and all those things. I have done my level best. If you put your level also, like your hand also, we can make this paper into the complete one. In the entire India, we are going to be the number one in making the things exactly however it is there in the pattern, right? I'll be grateful for everyone. If you want, I will also mention every one of your name, no problem. Okay. The next one, important notes, as I mentioned. Which one?
Sir, can you just go 2 slides back, sir?
Yes, please.
Just for 1 second.
Yeah, please.
2 slides back, sir. One more. One more. Before this one, sir.
This one?
After this, sir. After this.
After this. Okay.
No, after this.
This one?
After this, sir. 115, 116, from 116, sir. Yes, sir. Yes, sir. This one. Thank you, sir.
Anyway, I'll be sending this soft copy. Go through and let me know if there is any corrections. Okay. Please, put your hand also.
Sir, what was 135, sir? Sir, what was 135 questions, sir, actually?
I'm not sure. That's what I'm telling. Sexual reproduction method, something like that will be there. I'm not sure. That's what I'm telling. See, like in the 150 questions, they have sufficiently.
Recombination, heterokaryosis, something.
Sir,
Yeah.
135, it was something like spermatization and gamete-angel contact kind of thing.
To answer this question, we must know what exactly the question is, because without question we cannot say what exactly it is. That's what I'm telling. If you remember, you try to remember it. Just imagine, in this 150 questions, we have sufficiently equivalently discussed it to 1500 questions. That much questions they have given. But anyway, no problem. I think I'm running out of the internet. Anyway, just see, these are all the important ones. As I told you already, there's a disclaimer once again. Can you hear me, right?
Sir,
Yeah.
sir, sorry to interrupt you. A bit, sir, based on your exam and experience, what is the minimum cutoff mark for general and OBC candidates?
50. Mains, probably 50 to 60. In between. See,
Sure, sir, sure.
probably, based on this toughness, I'm expecting in between 50 to 60. Because the question is very,
Okay,
like medium range, right? Probably I'm expecting 50 to 60. If you are in the range of 55 to 60, I would say, you are safe for ARS mains. We cannot say, in one of the case they have given cutoff probably 45, something like that. Beyond 45, everyone can eligible, something like that they have given. But this time we will see, probably if you are above.
Sir, if a candidate is above 60, he is safe or not?
100%, there is no doubt about it. Mostly you are safe if you are above 60. So anyway, we'll see. Hope for the best. We'll see. As I told you, we cannot predict what they are going to, set a cutoff. But we believe this is the, based on my experience over the years, if you are above the 60, you are definitely safe. There is no doubt about it. Let's see. Hope for the best. Important, as I told you, these are all pure disclaimers, a couple of the optional things. Yeah, your reactions in the exam. By seeing the question paper, my first reaction, what the hell this question paper is, right? How many of you feel this one? Every one of you?
So all of above.
All the above. That's right, all the above. Another thing, this is for you only. Examination may answer.
So my reaction was, what the hell is this? this this kind of arrangement.
Exactly,
Mouse is not
everyone's same.
working and
See,
things are not
see in the examination, you thought that this was just before only I read. Yeah, I can't get it. So another reaction, right? There will be another couple of reactions for you.
Yes, sir. True, sir.
Yes,
It is
true.
It is with me, sir.
This is with everyone. The next one, mind reaction when I try to
Sir,
stuff.
I was like, nah.
Yeah. Yes, please.
Sir, I was like, every question was like a 4 marks answer, sir.
Yes, exactly. They have given that much, right? Anyway, another meme for you.
So this meme is very great.
So mind's reaction when I try to stuff in the content in the big books, no means no. No. Okay. The next one. This is a normal thing. It will never encounters because this I placed it because my examination center was very tough. Actually, yes. Actually, they asked me to remove my belt also. My pant is very loose here. If I remove my belt, my pant will go down. What I do? See, this is my reaction there. So the next one, when this is applicable for everyone. When the question paper is so brilliantly set that you become speechless, answerless, contentless in the examination hall, this is how our reaction will be like. Yes or not?
Yes, sir.
Yes, sir.
Exactly, right?
Yes, sir.
Two last memes. Syllabus, exam, student. This is the case one. Coming to case two. Me in the exam, what we think? Me thinking I am going to make it, I am going to kill it by seeing the question. This is how it will happen. Right? By seeing the questions, you will realize it what it exactly is. Right? One last thing, okay? This is the present situation of every one of us, right? The situation of some
Yes,
student
sir.
right now. This is the right now, but there is nothing to take serious about it. Okay.
Absolutely correct, sir.
Yeah, this is the last one. After writing a 2 hours of exam with 150 marks, you feel that I'm still alive. Is I'm still there? You feel like, right? One last thing. After exam, see how bad it is. Not a big deal. But who cares? At last, the exam is finished, right? The exam is finished. No more net this year or
This
maybe
was
no.
me, sir.
This is me, sir.
Yes, every one of us, right? One last thing. This is a serious thing. See,
So to forget some of the
yeah, tell.
Sir, actually, when I came outside, I thought paper was so easy, so I was happy. When I entered to the my bus or train, then so many I am thinking, oh, this also wrong, this also wrong. Like that what happened.
So
Till now,
see,
already so many wrong.
yeah, that is not the case with you. That is also case with everyone, including me. But I was very careful in answering because I only constructively answered few questions. But of course, it will happen with everyone, including me. I'm not exceptional. Okay. Another
Sir,
thing. This
sir,
is
how many questions you attempted, sir?
I attended only 120.
Yes, sir. This is very
Only
important
one.
question. How many questions you have attempted?
I have attended only 120. Only 120.
Huh?
Only 120.
120.
120 because
Okay.
you know
Great, sir.
there were few things. Yeah.
Mere 120. You have attempted 120 means what is our case, sir?
No. That is not the case. See, that is not the case. See, for
62
me,
is in our case.
no. That is not the case. See, I'll tell you one more thing. See, for my case, for me, it is just experience to let the others know in the upcoming generations. So, in order to, if I want to tell, I need to have the experience. For me, I'm writing experience. For net, I have finished in 2018 itself.
Sir, 50 to 60.
Come again?
Sir, whatever you are saying, 50 to 60, that is person, no? For means.
No. See, 120.
Yes, percent.
Yeah, definitely. That was percent.
Yeah, that's what I'm telling.
Marks
See,
because 75 is needed to clear.
yeah, 75 questions I'm telling. See, there is nothing to know. See, be cautious, okay? See, for me, it is just an experience. I'm writing just in order to tell the upcoming generation, it is just for experience. For me, I have cleared net many times earlier. Now, for me, it is just an experience. Even though I can hit 150, it's not a big deal for me. Even though I go negative marking, no problem. So, I will be telling one more thing by saying this one. There is a one case where I have my examination marks was 49.98 out of 50 because I am comes under general category. My percentage is 50. You just see how I might have feel like just ending in 49.98 or probably 97 somewhere, and probably in some cases. Just imagine how I feel. I feel very happy because it is not about how many times we are clearing. If you are not clearing, how many ways you are learning, that is the point here. If you see this is a serious things. You can see this is just one example. There are hundreds of examples are there. Can you hear me right?
Yes, sir.
Yeah, this is the case of Michael Jordan. You know, it is one of the greatest what was that, basketball, right? Is one of the greatest basketball, right? So in his career he have missed a lot of things, all 9,000 shots, 300 games, so many things would be there. This is one example, which means, so if you ask me, I'm not also an exceptional. Of course, I have also struggled in the initial days of my career. I'm telling you, take it as an experience. In the upcoming days, take it as an experience, like a sweet slap. So in whatever the way they are asking, I can tell. I'm telling you, majority of the time answers will be there in the questions itself if you read carefully. Of course, I can obey you. There is a problems like mouse, other things. Of course, other things will be there. So you need to 70 to 80 will be definitely in the list, like in this particular category also you can attend at least 70 to 80. So not this year, every year. I told you already, we are all soldiers. We are prepared for a war. Asking a king that, like, whether the war is going to be easy, no, it is not going to be easy, right? Definitely it will be difficult only. So this is what happened this time. Take it as an experience, hope for the best, don't worry about the options or the answers. However, however you have done the mistakes, in same way I have done the mistakes in the earlier days also. Even in this exam also I have done the mistake, but I restricted very less, because since I was very concise in selecting the answers and things. See, don't compare with me, because every one of us are individually different. Who knows, maybe you may got more uh, uh, you know compared to me, because I'm not in the line of preparing all those things. If I was in your situation, I may tell you I'm in the full track. Now I'm not in the full track, because I'm my thing of attending every single is just updating the knowledge. But anyway, forget about all those things. I hope everything will be fine. We'll hope for the best. Now, what are all the things you guys need to convey me? I think this is not the time, because I think the internet is ran out and this time is already too late. We do have a class, one more class tomorrow. We'll discuss all those things tomorrow.